incognito. Hello everyone, very good evening. I hope my audio and video is clear to you. Just let me know in the comment box. Alright, so from my side it is pretty clear. The audio video is going fine. Just let me know in the comments. If it is fine, we can start. Hello incognito, congratulations and welcome to this noble field, and I'm sure that you're going to be proud on your decision, except few times when you have to study more, right? Hello Unisoul, hello Ritvik, good evening. So everything fine bacho? Just give me thumbs up, nod, something like that, so that we can proceed. One shot physiology for INICET, it is planned bacho, and don't worry, it is going to be there on the Cerebellum app, right? My session is probably at the end of November, beginning of November or at the end of September. Medicos Ali, hi. Upcoming classes, yes, you can join the Cerebellum Insta page or Facebook page, right? So you can join there and all the schedules are going to be posted there, right? So you can go and watch the official page of the Insta, right? So yes, I'm good, waiting for all you guys. Yes, so let's start. Thank you VFS and Radhe, right? So you can go on official pages and you can know the schedules there, right? So first of all, we are going to start with a wonderful message, right? Especially to those who got the admissions in MBBS. Yes, definitely there will be times when you are going to be very proud on yourself, and trust me, at the end it is going to be worth it. Whatever decision you have taken to come in this field and serve the humanity, right, it is going to be worth it. But understand, sometimes the feeling will come that why I have chosen this field, because understand that your friends who have chosen the engineering or other fields, they are going to start the career paths very soon, start earning very soon, but trust me, they're not going to, you know, have that confidence to treat themselves, for sure, right? So not all positive changes feel positive in the beginning, okay? So whatever little we can do, we will do it, right? So I love the Big Bang Theory, and that's why I have put this meme over here. We are going to start the respiratory system, right? Whenever someone is going to say that love is in the air, so you have to say that no, the nitrogen, oxygen, carbon dioxide, they are the main ones in the air, right? So hello, good evening Madhivan. Roy, hi. Roy is here, nothing to fear, right? So you can help the incognito also. Right, so Weber's classification, that is very important question, not only just for the profs but also for the entrance examination. Many a times the question have been asked on this, right? So Weber was the scientist who gave his life to, you know, find out this or standardization, for the standardization of airway classification. Otherwise the CTVS people had a lot of problems to understand or have the surgery. If the patient is getting shifted from one place to another place, then they had the problems, but it is solved by the scientist, that is Weber. Good evening anonymous. So let's talk with it. First of all, understand there is a dichotomous division which is going to be present. For example, my drawing is bad, but still I will try. That you do have roots first of all of a tree, right? Then there is a stem, right? And those stems are going to divide into the main branches first, right? And those main branches are going to subdivide into further branching system, isn't it? And then those further branches are going to divide into many sub branches, and then there will be leaves, isn't it? So that's how the trees are going to be formed, isn't it? And in the childhood we used to make it like this, isn't it? So same system, you just reverse it, it is going to be your respiratory system. So that easy it is. It's just that it is not unorganized like a tree, right? It is well organized, and please remember it is the dichotomous branching or dichotomous division which is present in the respiratory system. What do you mean by dichotomous? Di means two, and understand, when I'm saying two, that means every branch will divide into two. It will not divide into three, it will not divide into one, or it will not just continue like that. Please understand, every branch will divide into two. You can see over here there is a proper organization which is present in our system, that is dichotomous division. So you can see this is the trachea, which is the stem. This acts as a stem for our respiratory system. From the nasal cavity, it is going to go back into the laryngopharynx, and then it is going to continue into the trachea, which is also known as the windpipe, isn't it? And then there is a dichotomous division over here, then again dichotomous division over here, and these branches are going to continue to divide continuously into dichotomous division. So please understand, whenever this is going to, this Weibel's classification is going to come as a question, please make a note. If you are writing a prof, mention this dichotomous division, because this is only written in the Gnome, and the examiner will know that you are definitely have read the Gnome, isn't it? So you'll be having a good impression on the examiner. Hello Dr. Amazing. It is for everything, bacho. It is for everything. If you are free right now, just attend these classes. I cannot say that this is not important for anything, because respiratory system is the most important system, right? Everybody, I'll say, if you have time, so you can go with that. Otherwise, you can just solve the PYQs for the entrance examination. At the end, I'm going to discuss some questions, clinical question I have arranged for you guys, which are definitely, I have taken those questions from the NEET PG or the previous year questions from the AIMS also. Okay, so upper, middle and lower. Now understand, yes, this is the, first of all they will divide into two, and then again they are going to divide into two. Okay, Roy? Yes, there is, but not always. Okay, let's move on. So what they are going to ask you is to jot down this Weibel's classification. Then what you are going to say is the trachea will start first, and then there is a dichotomous breathing division. Now trachea, there are 23 generations which are going to start from the trachea. So in total, Weibel said that there are 23 different generations. Generations 23 from 0 to 23. That means in total we do have 24, but we are saying 23 because we do not count the trachea in it. It is a number 0, right? So trachea is going to start this tree, and that is going to be generation 0. And remember, T to T is the conducting zone. I'll write that later on. Just you have to remember that T to T, that is terminal bronchiole, is going to be generation 16, right? Generation 16 is going to be terminal bronchiole. Then there is going to be a transition which is going to occur. What is the difference between that? Just understand, and then I'll make you write. So see over here that this is the trachea, isn't it? And terminal bronchiole. Now what you can see over here is that there is one division from here to here, and there is another division which is coming from the transitional bronchiole towards the alveolar sac. Now what is the difference between these two? If I divide it over here from 0 to 16, and then I'll continue the another section from 17 to 23. What is the difference is that the first 0 to 16 generations, they are just for the conduction. They're not for the transport. Now understand, if you went to some party and there are some chefs which are preparing or cooking the food, and there are waiters which are going to bring the food towards you. If the waiters start to eat up all the food, definitely the guest won't be getting anything, isn't it? So that's why the waiters are not allowed to eat, at least till the guests are there, right? So what they are going to do, they are going to conduct that food towards you. Okay, so and you will have that exchange. You will keep the empty plate and you will take the whatever dish you like, right? So similarly this is going to happen in the lungs also. So understand the air is not going to get exchanged unless and until it reaches to the respiratory bronchiole, and that's why it is known as respiratory bronchiole. Understand, as soon as it goes to the terminal bronchiole, what is going to happen, that it is going to go in the respiratory bronchiole and then it will start getting exchanged with the different pulmonary vessels, whatever blood that is coming there. So with those different gases, it is going to get exchanged. Okay, so there are two different zones available over here. First of all, from the trachea till the terminal bronchiole. T से T याद रखना, terminal bronchiole. You do have only conduction and no exchange of the air, and that's why these are known as conducting zone. So this zone over here is known as conducting zone. So if they ask the generation, tell us the generation about the conducting zone, then you will be saying it is from 0 to 16, right? See, sweet 16, they don't exchange anything, right? They just are dependent on the parents. So please remember, over there also this is only for the conduction and not for the exchange. Okay, from the respiratory bronchiole, this terminal bronchiole, they are going to end up into the respiratory bronchiole, right? So from the respiratory bronchiole, as the name suggests, the respiration actually starts over here. So respiratory bronchioles to the alveoli or alveolar sacs. What are the alveolar sacs? They are grape-like structure which actually collapse and not totally collapse, but they will just recoil and expand. That is going to be the more appropriate terminology. What they do is they increase the surface area so that the diffusion of gases with the pulmonary vessels occurs very easily, okay, and very quickly, right? So they are the grape-like structures. Now we have already mentioned that these are going to be 23 generations. So from this is the gases exchange are going to occur over here. So this is known as transitional zone, or this is also known as exchange zone, or this is also known as respiratory zone. So as you guys have the nicknames everywhere, so this zone is also going to have three different names. That is, it is either transitional zone or the exchange zone, because the exchange of gases occurs, and that is the primary function of the respiratory system, and that's why we call it as respiratory zone, which is going to be from 17 to 23. 17 to 23. I do not have to mention that respiratory bronchiole is going to be the generation 17 and this is going to be the generation 23. Okay, at least remember these two zones and their different. Okay, we are planning Aladita, we are planning for the full course, and full course is available. I just cannot tell you right now when this will be planned. This is just for the important topics that we are covering right now, but Cerebellum is definitely planning for the whole full course, in which we are going to start with the beginning and we are going to end it with the all topics covered whichever are important, right? So first of all, just focus on this class right now, right? I'll answer all the questions at the end. So you can focus that, at least remember the few things I want you to remember. The first thing is over here, that is the number of generation number of trachea, terminal bronchiole, right? Then transitional bronchiole. I know that the transitional bronchiole is going to start at 17, but 17, 18, 19, three things I want you to remember, that these three are going to get included into the transitional bronchiole if you can. Then it will be continued in the alveolar sac. So 3 you have to divide into that, right? So next three, that is 20, 21, I'll change the color. That is 20, 21 and 22, they are the alveolar ducts. And finally, as we already discussed, the 23rd generation is going to be alveolar sac. They are the grape-like structure which increases the surface area, right? Gold medal बिल्कुल जीत सकते हो बच्चों आप, बिल्कुल जीत सकते हो. There is no doubt about it. Okay. Now the next thing over here is the sizes of the airway. We know that there are three different sizes of the airway. First is large size airway, then the medium size airways, and then the smaller size airways. Okay, now understand, when we say large, medium and small, they are going to divide also, but they are parallelly arranged. So what is going to happen? So look over here. So if I say, as we discussed in the CVS system also, that in comparison to aorta, the capillaries, there are billions and billions of capillaries which are arranged in the parallel manner. And what happens to their resistance as you go forward? Though one single capillary area is very, very small, but if they are arranged in the parallel manner and you check out their cross-sectional area, which is going to be much more as compared to the aorta. Not only that, if you calculate the resistance which are arranged in the parallel, it is going to be very, very less. Many capillaries, as many as capillaries they are arranged in the parallel, that much lesser the resistance will become. Okay, and that's why we do have better exchange at the time of when the blood comes towards the capillary. So same is going to happen over here. Suppose this is two small or medium size airways, but they are going to divide into multiple branches, isn't it? And then they are going to make many small branches like these, many small branches. So if you take their cross-sectional area, overall cross-sectional area is going to be much more as compared to the cross-sectional area of the medium size airways. Now understand what is going to happen to the speed. When you do have less, you can say, traffic, you go in a more speed. Same is going to happen over here, since the speed which is going to lesser down in the smaller airways, because the intraluminal space is less there. Over here in the medium size airways as compared to the smaller size airway, the speed is going to be much higher. But understand, when you go with the slow speed, you do have more control over the air or more control over the vehicle that you are driving. Same is going to happen over here. If the lesser speed is there, that means there is a proper time for the air molecule to form a laminin, isn't it? And that's why the flow is not going to be turbulent or it is not going to be noisy, which is going to take place in the case of medium size airways. Okay, the phenomena I wanted to talk about was the resistance. First of all, that if you arrange, let's say I'm just rubbing this off, or I'll just write it on another paper. So if I say that let's say this is the resistance which is arranged in the series manner, R1, R2 and R3, isn't it? And there is another scenario in which I'm going to attach the resistance in the parallel manner. Let's see what happens there. So let's say this is R1, R2 and R3. R1, R2 and R3. Now understand, when I combine all these three, what is going to happen? When anything is attached in the series manner, you just calculate R1 plus R2 plus R3, and that's how you are going to get the total resistance. I'm going to write it as T, right? So suppose this is 2, 2, 2. Over here also it is 2, 2, 2, right? So if I say the unit is 2, or there is a resistance of that, so what is going to happen? 2 plus 2 plus 2 over here, in this case it is going to be 6. But when I'm going to calculate that in the case of the parallel manner, R1, R2 plus R3, this will be like this, right? First of all, we have to calculate the R dash, and then we can calculate the RT in total, right? So it is going to be 1 upon 2 plus 1 upon 2 plus 1 upon 2, that is going to be 3 upon 2, isn't it? And if I have to calculate, but this is, understand, 1 upon R dash, okay? So if I have to calculate RT, then it will become reverse, that is 2 by 3, right? So 2 by 3 is going to be the answer, right? and this is going to be around somewhere around 0.6, I guess, right? So 0.666, like that. So this is going to be the case. But if I add the resistance over here, see what is going to happen. I'll change the color. So if I add the resistance, this is going to add by 2, this will become 8. But if I add the resistance in the parallel manner, what is going to happen, that I have to add one more over here. So this will become now 4 by 2, isn't it? 4 by 2, and this is going to be reversing in this case. So I'll just write it over here. 4 by 2 will be the 1 upon R dash, and RT will be, in this case, the RT will be 2 by 4. That means it is going to be around 0.5. So you can see the resistance is rather decreasing. So same is going to happen whenever you are going to attach many smaller airways in the case of the respiratory system. So same over here with this case, we can move ahead that the cross-sectional area of medium-sized airways as compared to the small size airways is going to be lesser, right? And this is going to be more. Velocity, understand, on the other hand, because of the intraluminal space available, the velocity is going to be higher over here, and this is going to be lesser as compared to the medium-sized airways. What about the Reynolds number? I told you that the Reynolds number is same as the velocity. It is directly proportional to the velocity, and we know the more will be the speed, lesser will be the control, and that's why the higher will be the this Reynolds number, and over here it is going to be lesser. And Reynolds number depicts the turbulence. That means how noisy the flow is, isn't it? So the sounds are going to be there, right? Absolutely true, Roy. Yes, you are correct. So this is going to be the case, and if the Reynolds number is more, which is the number for the turbulence, which is showing the turbulence, how effective the flow is. So understand the turbulence is going to be much more in the case of medium size airway, and it is going to be lesser in the case of the small size airways. And understand, lesser the turbulence, effective or efficient will be the flow, and that's why it is the smaller size airways which are going to exchange more of the gas, right? So this is the case, and remember the maximum resistance is found in the medium size airways, not in the large size airways. Which resistance I'm talking about? I'm talking about the airway resistance. Okay, so I'll write over here maximum airway resistance, the site of maximum airway resistance, because they are going to, I'll show you some questions at the end. They're not going to give you direct question. Definitely they are going to give you the indirect question. Understand, many universities are giving one-liner question, but in Delhi University especially, they are not going to give you one-liner MCQs. They are going to give you the case scenario, and you have to find it out. If your concepts are strong, you are going to able to do that. First of all, let's see why it is the medium size airways and not the larger size airways. If I compare these two, if I do the same comparison in the case of medium size versus larger size, then the larger size airway should have more resistance and they should have higher velocity, isn't it? But understand, they are extrathoracic. Mainly the larger airways like trachea and all, they do contain cartilages, plus they are extrathoracic. So whenever you are going to expire, the positive pressure will not be applied on these airways. Okay, so because of these two different points in the large airways, these are not the site for the airway resistance. When the resistance is going to come, whenever there is a obstruction intraluminally which is going to be present, which is not there in the larger size airway. Mainly they contain the cartilages, mainly because they contain the cartilages, and they are extrathoracic. So whatever thoracic pressure changes are occurring, they are not affected by that, and that's why they cannot be the site of or main site of airway resistance. So who is the main site of airway resistance? This is going to be the medium size airways. Okay, and if we discuss, this is according to their cross-sectional area and their speed, but if we discuss that according to the volume, what is going to happen? So understand, whenever, for now just remember, whenever I inspire, what is going to happen? The negative suction pressure inside my lung is created, and that's why I suck the air from the atmosphere, right? And whenever I'm going to expire, what I'm doing is I'm going to put a positive pressure. How this pressure is applied and how it is generated, we are going to learn everything step by step. But for now, just trust me that I'm telling you correct, that whenever inspiration is going to occur, we do have the suction or negative pressure. Because of the negative pressure, what is going to happen, that the airways are going to widen up. And understand, whenever you are expiring, what is going to happen, the air, they are pushed and the alveoli, they try to recoil, and because of that, what happens, the narrowing of airways will take place, and because of that, the resistance in the airway, that is also going to change. When I'm inspiring completely, that means I'm at my total lung capacity. At that time, the resistance is minimum. So according to volume, if I talk, the minimum resistance is present at the TLC, that means total lung capacity. Total lung capacity. And what about the maximum? It is going to be maximum at the RV, that is residual volume. That is going to be the residual volume. Okay, that means whenever I'm going to expire the max, that means forceful expiration, at the end, whatever amount of air remain in my lung, that at that point I'm going to have the maximum airway resistance. Why? Because the airways are already collapsed. Whatever are present inside the thorax, that is known as intrathoracic airways, right? So that is the case over here. Let's move on to the next thing, right? Oxygen coming through the respiratory system, but yes, it finds the way towards the circulatory system, right? We do the enjoyment and everything, and later on we realize that we do have less time, so we find out some shortcuts and still we get through the exams, right? That's how the life is, and we are also shocked that we have reached this stage, right? So same, as much as shocked as him. Okay, so let's move on. Respiratory functions. What are the respiratory functions? Just answer me, what is the respiratory function, and then we will learn about non-respiratory function of the lung also. Just answer me, what are the respiratory functions? Two main respiratory function, and then rest of them are going to be the non-respiratory functions of the lung. Chalo, answer fatafat se. We'll move on forward. Chalo, what is the primary function here? What is the primary function? So many people are watching, but nobody is answering. You know, कि मैम आप ही बताओ. Very good, right? So yes, this is the main function of the respiratory system, to respire. Respire means to inspire and expire, right? So first thing first is the gas exchange. Understand, most of our energy comes from the aerobic metabolism, and for that we require oxygen because it gives maximum energy and minimum expenditure, isn't it? So it is the best economical system possible for the energy extraction, and that's why we are going to have what thing, the aerobic respiration, and for that we require oxygen. So exchange of airways, gases through the airways, that is going to be the main function. We take up the oxygen and leave the or expire the carbon dioxide. Not only the lungs require the oxygen, but also all the system, and that's why we have to send it into the blood, and through the blood it is going to go towards all the organs present in our body. Aerobic metabolism you can write, and that is going to give us the ATP. Aerobic metabolism, which is going to give us the ATP. Now tell me, we control the carbon dioxide output also. Sometimes we hyperventilate. Sometimes our ventilation, our respiration is slower. Now, why are controlling this carbon dioxide output? What we are achieving over here? Yes, they are the non-respiratory function, Roy. You are right. Yes, these are the functions, but again, these are the non-respiratory function. You have told just before this the buffer system. Yes, that is the respiratory function via controlling the carbon dioxide output. That is the function our respiratory system is doing for us. Now, via controlling the carbon dioxide, now understand the carbon dioxide is the volatile acid. Volatile means we can excrete it out very easily with the help of gas, right? But this is the acid. If there is a acidosis, what we should do? Acidosis means there is high acid in the blood, right? So we have to manage the other acids. So what we do is we excrete more and more carbon dioxide out, so that in the blood less carbon dioxide will be present. If less carbon dioxide is there, more will be the bicarbonate. Okay, so via this volatile acid, what we are controlling in the blood is that in blood we are regulating bicarbonate or serum bicarbonate levels in the blood. Now understand, the bicarbonate itself is the alkali in the blood, right? It's the base in the blood or alkali in the blood, right? So it is not the acid, but if the carbon dioxide is not there, there is no one to bind to the bicarbonate. Okay, otherwise what will happen if more of this carbon dioxide is present, then the reaction is going to go in the other way with the help of the carbonic anhydrase, right? So via maintaining this, we are actually, we can say this is base, or we can say that this is the main buffer, main or major buffer everywhere except few places, but major buffer at least in the blood, ECF, whatever they ask, this is going to be the main one. Okay, so this is the main buffer which we are controlling with the help of our volatile acid output also, right? So this is the main function of the respiratory system, right? If the lungs are there, the oxygen and carbon dioxide, they just jump in and jump out, right? So I got this meme, I found it very funny, right, when I saw it, so I just added over here. Let's move on. Now tell me the non-respiratory function. Some of it I got it. Yes, epiglottis is closing when we eat the food. Yes, but that is not per se occurring via the respiratory system. Epiglottis is the different thing from the respiratory system, right? Yes, order it is there, cough, spasm, alright, that is there. So to avoid the aspiration. Aspiration means the thing or the fluidic content that we do not want to go inside the our solid or the fluids, they should not go in our lungs. Okay, yes, that's also correct. Fine, so let's talk about one by one. First of all, remember that the pulmonary vessels, I told you that they are going to take up whatever. I told you in the CVS system that the auto-regulation of the blood is absent in. There were two things in which the auto-regulation was absent. What were the two things? First was the skin and the second was the lung. Lung cannot control how much blood they are going to get. Whatever blood is coming into the right atrium and right ventricle, it is ultimately going to go in the blood, pulmonary system, right, via the blood. So what is going to happen, that the cardiac output of the right ventricle, the whole cardiac output of the right ventricle, it is going to go in the lungs or the pulmonary vessels. So lung should have high compliance. Compliance means the stretchability. We have discussed that already. Stretchability or distensibility. Stretchability or distensibility, that is going to be higher in the case of these vessel. And I told you that if someone is having higher compliance, they can stretch or they can distend. If they can distend, that means they can store the blood and they can control how much output should be going towards the left atrium, left ventricle, so that at a time more load will not come on the heart. But whenever more blood is required, more oxygen is required at a time, for example at the time of exercise, they are going to get compressed, and that's how you're going to get the more blood which is not there at the resting condition. So via acting as the high compliant vessels, the pulmonary vessels, they do the function as the reservoir, or you can write temporary reservoir, because the whole cardiac output of right ventricle, it is going to go to the pulmonary vessels, isn't it? And that's why it acts as a temporary reservoir of the blood for left ventricle or left heart. That means LA and LV, the left atrium and left ventricle. So for these two things, it is acting as the temporary reservoir. So that is the first thing. Yes. The second thing is filtration. What they are filtering? They are filtering many things, right? First of all, you must have heard of PM. What is PM? This is particulate matter, not the PM that I'm talking about, right? So they are going to filter many things, right? From the air also and from the blood also. From the blood, what they are going to filter? Various things are going to be present in the veins. For example, if someone had surgery, right, just a few hours after that, the clots, they can get breakdown from the site of the injury or site of the surgery, and they can come into the veins, right? And via the venous system, via the venous return, it is going to come into the right ventricle, and they can go into the left ventricle, and via the left ventricular cardiac output, it is going to go in the microcirculation of brain, heart and everything else. So that's why we do not want them to go there. So hence, these filtration of this pulmonary vessel, they are going to act as a shield for all those microemboli, air and fat bubbles which are going to come towards the this right heart, right? So filtration of microemboli, the fat emboli, right? Sometimes the fat globules, small fat globules, they can also come into the venous system, right? And through that they are going to come into the right ventricle. But since the pulmonary vessels, they can have this filtration system, what they are going to do is they will not allow these microemboli, fat globules, small fat globules or air bubbles to enter into the left ventricle, and so that they are protecting our vital organs and their own capillaries, vital organs capillaries such as cerebral capillaries, to not get blocked by these things, right? So which are going to come from the filtration of microemboli, fat and air bubble, which can come from venous system. And if they go in the arterial system, what is going to happen? Definitely they are going to go and block the small vessels of the vital organs. If they block the small vessels of, let's say, cerebral vessels, the small vessels of the brain, what is going to happen? The blood flow won't occur to that area. And if it is the end artery, for example, that means, what do you mean by end artery? That means there is no another supply for that region of the brain rather than that particular artery. This is the only branch which is supplying the blood to that region of the brain. What is going to happen if that artery is blocked? Definitely there is no one that can supply the blood to that area. That area is ultimately going the ischemia, that is lack of the supply of the blood, and then it is going to go in the necrosis condition. That means the death of that tissue is going to take place. And if the cell death occurs, definitely whatever function they are doing, there is no one to replace that function, especially in the CNS, right? If that these embolize, fat molecules, air bubble, they go in the heart, what is going to happen? The heart circulation will stop, and this is known as the heart attack, or commonly known as heart attack, that is the MI, right? Myocardial infarction will occur, and many places this can go and cause the necrosis like severe conditions also. So thanks to our pulmonary vessels, smaller capillaries especially, that they filter out these things and they just destroy it with the help of PAMs, that is the pulmonary alveolar macrophages. They are going to digest it and they are going to excrete it out of the body. Next is the biochemical function. Now, what contains in the biochemical function is that they are going to synthesize some hormones, they are going to synthesize many other things, they are going to release many things. So what are the things that they release? They secrete. First of all, bradykinin, then the bradykinin, leukotriene, leukotrienes, then serotonin, bradykinin, leukotriene, serotonin, right? Then comes the prostaglandins, various prostaglandins such as prostaglandin E1, prostaglandin E2, prostaglandin E2, and prostaglandin F2 alpha. F2 alpha also getting released by this. Okay, not only that, there is one more molecule which is the kallikrein, which is also synthesized and released by this lungs, right, which are going to help in the various conditions, especially the inflammatory conditions and all, right? And as we are going to move forward, we'll see their functions. Yes, Roy, you are giving me the answers. Very good, bache. Very, very good. If this question comes, you should get the full out of full marks, right? The next function we are going to move towards that. The processing of air is going to take place, especially in the cold areas. What happens is the air gets dried up, okay? And if the dry air comes in our lungs, what is going to happen? That irritates our airway system. And if the irritation is there, allergic conditions or inflammatory conditions, they are going to provoke. And they are going to block our airway system, which we do not want. And that's why, whom do we have to protect from this nasty dry air? We do have cilia. What they are going to do? They are going to secrete the mucus, moisten that air. Not only that, but they also control the temperature of the air. That means whatever temperature inside the body we are having, the air should be that warm. Otherwise, again, that will cause the irritation, right? So that's why the processing of air mainly occurs in the nostril, that is the nasal cavity, and our larynx, trachea, wherever the cilia are there, right? So thanks to the cilia, this is going to not have the way in our lungs, right? So respiratory system is prevented. Now, next, non-respiratory functions. First of all, delivering the odorants. In the nostril, we do have the receptors for the odorants. What is going to happen? As soon as we smell something or we take the air inside our nostril, there are odorant molecules, that means which are going to have or give that smell property to that particular substance. They are going to get trapped in our nostril. Okay, and because of that, what is going to happen? Once they are trapped in our mucus, they are going to taken up towards the receptors, olfactory receptors, which are the bipolar neurons which are present, right? And they are going to grab that odorant molecule and process on it, and will get to know whether it is a sweet smell or a pungent smell or what type of smell it is. Okay, so the olfactory function, that is a sense of smell, that is also going to come from, sense of smell is also going to come from the nostril mainly. And first nerve, that is the olfactory nerve, right? Then coming to the processing of inhaled air. What is the processing that is going to happen? First thing is the air will be warming and moisturizing of air. Warming till what point? Till the time the air gets to our room, this body temperature, right? So that's why if there is a cold environment and you start expiring, you'll see that your air that is coming out from your body, that means coming out from your nostril, it is warmer as compared to the outside environment, right? Why is it so? Because it is warmed up by our nostril and respiratory system to our body temperature, so that it will cause least irritation to our body. Okay, so that is the case. First of all, filtration is going to take place, then the warming, and then the moisturizing. So for the moisturizing, we are going to sprinkle the water vapors to it, so that it will get moist and that will not cause the irritation as it can happen with the dry air. Okay, and that's why in the cold you must have seen that the asthma patients are started to have more attacks in the winter. So why? Because the dry air will cause irritation, and mainly if they are dehydrated, definitely it will cause the more severe attacks, which is because of the lack of moisturization of the air. Okay, coming to the endocrine function, they are going to secrete the three main things, that is the ACE. What is ACE? Angiotensin converting enzyme. As you guys already know from +1 to +2, if I tell you a very simplified diagram, what happens is, first of all, there are three organs which are going to work in the harmony. What are the three organs? First is the liver, then is your kidney, and the lungs. Right. Now understand, the liver makes the protein which is known as angiotensinogen. And I told you angiotensinogen, ogen, wherever the ogen is going to come, that means it is inactive. We have to make it active. And for that, whenever required, the kidney is going to release renin. The renin will come towards the angiotensinogen and it will convert it into the angiotensin. But remember, it is still not active, and that's why we are going to call it as angiotensin 1. Then what will happen to this angiotensin? Thank you. This angiotensin will go towards the lung. Lung are very happy, right? What they are going to do? They are going to release something which is known as the angiotensin converting enzyme. From where? My question to you guys is, from where these angiotensin converting enzyme has been released? Are they released from any cells? Are they released from the vessels? Are they released from the interstitium? From where these ACE enzymes are released, right? As soon as this ACE enzyme is going to come into the blood, what is going to happen? Definitely this angiotensin 1 will be converted with the help of ACE. It is going to get converted into angiotensin 2. There is angiotensin 3 and 4 also, but for now we'll stop it over here. And this is known as renin angiotensin system. Okay, so please understand, renin angiotensin, and this angiotensin can go to the suprarenal gland which is present, and that can make it release the aldosterone also. So that's why this is known as renin angiotensin aldosterone system. Okay, because this second A is for aldosterone. Type 2 pneumocyte, anonymous, think again, but I am not asking about the surfactant, I am asking about this ACE enzyme. ACE enzyme is released from which cells, or from the interstitium, or from any other thing that you have in the lungs? Yes, very good, Roy, absolutely correct, that these are released from, please remember, these are released from endothelium. As I always tell you, do as many as mistakes you want in the lecture, so that you will avoid doing those same mistakes in your exams. That is very, very important. That's why I encourage you to, do as many as mistakes, because whenever you do the mistake, that you cannot forget for your life, right? So endothelium, endothelial cells of the pulmonary vessels, from there it is going to get released, pulmonary vessels. So from the pulmonary vessels, it is going to get released, right? So please remember that, which are there along the alveoli. Okay, let's move on. So angiotensin converting enzyme is going to get released. With that, which are the two more things? I have the mnemonic for you. ACE the pH. What is ACE the pH? Obviously the prostaglandins will be released and the histamine will be there. So all these things will be released from there, right? And this is going to cause the changes, whatever endocrine changes are going to happen. Whenever we require aldosterone, whenever we require the angiotensin 2, this is going to help us out. Now what they are doing, they are inspiring and expiring these lungs, right? And they are against each other and they are protecting each other, right? So please remember, the another thing that they do is the defense mechanism. They defend us. From whom? From various harmful substances, from toxins, from the foreign particles. And you must have heard, especially in Delhi or any metropolitan cities, when they talk about the pollution, they talk about PM 2.5, PM 5, PM 10, right? And what are their levels? Now, what is this PM? This is nothing but the particulate matter. And what they talk about, the values, that is 2.55, that is the diameter of those particles. Why it is very, very important? Because understand, if they start rising too much, we are inspiring that air. Via the inspiration, they are going to go and lodge in our respiratory system. Our respiratory system is perfectly fine to defend against that, but if there is a particular level, right, till which it can fight, but after that, if there is an excess accumulation, definitely our defense system will give up, right? And various conditions will arise, like fibrosis will start and difficulty in breathing. Asthma patients will have more problems in this case, right? So all these problems will start to occur, and that's why it's very, very important whenever they say wear mask for your own health, right? There is a possibility, one research is going on, I have read that maybe that the future generations are going to have inbuilt filters, or there will be some mutation which can occur due to it. So either the mutation can go in a good way, that means we will be developing the tolerance against them, the particulate matter, or there can be diseases which can take place because of that. The particulate matter, the pollution is going to definitely cause the cancerous condition, so that is also important, right? So let's talk about it. The maintain the integrity of the mucosal lining. First of all, we'll talk about the particulate matter. Now understand, the particulate matters, usually we are going to divide it into the let's say more than 10 micrometers, 2 to 10 micrometers, and less than 2 micrometers. Now understand, as you go down towards the alveoli, the airway size, it will get narrower and narrower, right? And they do have cilia which are going to be towards the nasopharynx, okay? So we do have cilia in our nasal cavity also, we do have in the airways also. Both beats towards the nasopharynx, okay? So the nasal nostrils wale, they will, when we go towards backwards over here, so they will beat downwards, and the pharynx and other airways like trachea and all, they will beat upwards. So towards the nasopharynx, they are going to beat. And for that we require the molecular motor, that is dynein. With the help of dynein, the cilia are going to beat, okay? With that beating, first thing, because of the mucus lining is there, right? So the cilia and the cuboidal cells are going to be there, which are going to secrete this mucus because of the sticky material which is there at the lining of our airways. The particulate matters, dust particles, or any foreign matter for that matter, or bacteria, viruses, they are going to stick on that mucosal lining, and because of the cilia, that whole mucus will be excreted out, isn't it? So please remember, as you go down, the size is going to get narrower, and as soon as the air hits to your nasal cavity, it is going to hit with a lot of force, and that's why it cannot change its speed. And we do have conkers in our nasal cavity, because of which they will act as an obstruction, and they obstruct the big particles to go more forward, right? So if it is more than 10 micrometers, they are going to get trapped in hairs of the nostril. Hairs and conchae of the nostril, you can write. Nostril, that means the nasal cavity that I'm talking about. Nostril. Okay, so I'll just write nasal cavity, better word, medical terminology. Okay, now 2 to 10. What if they are falling between 2 to 10 micrometer? These micrometers that I'm writing over here, these are the diameter of that particle. Okay, very small particles in the micrometer. Okay, so that is the diameter of that. If that is there, they are going to go in the conducting airways. But remember, the conducting airways also has divisions, and they go in the different direction. Plus they do have cilia, mucosal lining, and because of that, these particles till 2 to 10 micrometers, they are going to trap into the conducting airways, and that's why they are not going to affect our exchange. So it is not going to harm us that much, right? But if they are less than 2 micrometers, what is going to happen? At this case, they will go towards the alveoli. Okay, so yes, they are going to go in the exchange zone or respiratory zone, and they might affect the alveolar gas exchange. But understand, we do have defense mechanism for that also. Our lungs are fully prepared army. Okay, for that what we do have 2 types of macrophages. First is the PAM, that is known as pulmonary alveolar macrophages or interstitial macrophages. Phage means what? Phagia or phage. Phage means to eat up. They are going to eat up those particles and do not allow them to block or clog the pores which are present for the gas exchange, and that's why the gas exchange will occur efficiently, right? What is PAM? Pulmonary alveolar macrophage. Pulmonary alveolar macrophage, right? So these are there in the exchange zone, and if the dust particle comes or the PAM comes, or even the viruses are very small, so if they comes, these are the armies which are present with their guns, right? They are going to eat up, right? And as soon as they eat up, definitely they are going to have lysosomes. They will destroy that particle, which is known as the digestion of the particles. Okay, so we do not call it as this thing. We do not call it as destruction, we call it as digestion. They're going to eat up, they're going to break them down in a smaller particles so that they can be easily excreted out from our body. Now understand, I was talking about the levels. Okay, if the air is having a particular level, it is healthy that we can fight against it. But if the level is too much, what is going to happen? These particles start to clog in our alveoli. If they start to clog, what is going to happen? Definitely that will cause fibrosis, and that will lead to, that means they will irritate, first of all, irritate the mucosa. That's why the fibrosis will start. And the second thing that they can cause is that they will clog the, suppose this is the respiratory membrane, they are going to clog, they are going to go in between and clog the pore through which the oxygen and the carbon dioxide was getting exchanged. What will happen if you obstruct the flow? Definitely the patient will land up into dyspnea or hypoxia like condition. So that's why it's very important to control the pollution, isn't it? Next, now understand the next thing, that they have this protease activated receptors also. What they are going to do when they are activated? When they are activated, they will release PGE2, right? So with the help of that also, we can work as the different biochemical structures will be activated with that. Not only that, the interleukins will be activated. So if is there any particle which is causing harm, we can fight with that. Okay, so this is kind of an inflammatory response which can be seen. Not only that, the interleukins are there, plus the mucosa lining. One more thing, whenever we do have mucosa, please remember that wherever we do have mucosa, it is going to have the antibody, the fighting system, the most important immune system, that is the immunoglobulin A is going to be there in all the mucosal linings for that matter, not only in the respiratory system, everywhere. In the mother's milk also we do have IgA. So secretion mein there is going to be IgA which is going to be present. Even in the saliva you do have, nasal mucosa you do have, right? So our army is everywhere. It's just that you have to keep it healthy via eating properly, via doing the exercises, via studying the medicine, right? So the mental health is also important. So everything adds up to one thing, that it is the best defense mechanism present in our body. Okay, let's move on to some more functions. What is the other metabolic functions that are present? Understand that they are going to cause the removal of something, they are going to cause synthesis of something. So what are the things that is going to get synthesized? So synthesis, they are going to produce something known as surfactant. They are going to produce something known as surfactant, which is going to be there, right? And once the surfactant is produced, what is going to happen? There is something known as surface tension which will be reduced. Surface tension will be reduced over here. Now what do you mean by surface tension and compliance? We'll read that. Okay, right. So we'll read that. Let's discuss rather going forward before that. So suppose, I'll just explain you in the simple diagram format. Suppose this is not a balloon, this is a, let's say this is an alveoli or alveolus, right? So alveoli is there. Now there are forces acting upon it. This alveoli is not just filled with the air, but also understand that there is a lining which is present. This is a water liner. Okay, so the alveoli, they are lined inside, they are lined with the water, because whenever we were in the mother's womb, that is we were fetus, so in that time what happens is alveoli are collapsed. There is a negative pressure which is present, and because of that the blood which is flowing through the pulmonary circulation, through that some of the water comes over here, right? And that's how they are going to get lined up, and that is going to be there forever. Now understand, we are inspiring air, and you must have known how the raindrop is going to have that shape like this. This is because of all the cohesive and adhesive forces that are present in the two medium and the within the water itself, right? Same funda is going to be applied over here. The water molecules that are present here, they will try to come together and form a big one single molecule. But what is the problem? The problem is that the alveoli will collapse. And if you start inflating the balloon, you will get to know that as the balloon is slightly inflated, it is easier to make it inflated more or inflate more air inside it, right? But if you start it with the collapsed state, from the collapsed state, it is much more difficult to fill the air. There is a lot of resistance present. Understand, if the same happens with our alveoli, what is going to happen? That if we start from the collapsed alveoli, every time we have to expand it from the collapsed state, it is going to be taking a lot more energy, and it is not at all going to be economical, and we are going to lose all of work done there, and definitely it is going to be a lot more resistance against which we have to act for. But if we keep some air inside it, it is much easier to expand the alveoli, to inflate the alveoli, and that's why we should find some solution to this. What is the solution? There is something known as surfactant which are going to be present. I'll just draw it in another color. So there are the thing which can float on the water. What is it? That should be less dense, right? That should not be altering the property of air or water, and that should not be harmful for our alveoli also. What is that thing? That thing is nothing but lipid. If you order any curry from the restaurant, for that matter, Indian restaurant specifically, then you will find the oil floating on it. As soon as you open the cover of that, you'll find a lot of oil in that. Why? Do they add oil after making the sabzi, curry, whatever it is? No. They have added it already, but the oil particles, the lipid particles, they are less dense, and that's why they come on the top, right? So same is going to happen over here. We are going to produce something known as surfactant. And they are going to oppose this force. The inward force that is present because of the water and air interface, that is called as the air-water interface. So, air-water interface, because of which whatever inward force is present or generated, that is known as the surface tension. That is called as the surface tension, which we are opposing over here with the help of surfactant. Surfactant is going to oppose the surface tension. That oppose, or you can write, it can reduce the surface tension, which is going to be helpful for us so that our alveoli will not collapse, right? One more thing that is going to happen is that something known as compliance. It is also a force which compliance is stretchability or distensibility, which actually act in the direction of surfactant. Surfactant will try the alveoli not to collapse. The compliance is working in the same direction. Understand, the surface tension is inward force, surfactant and compliance acting in the opposite direction. Yes, it is the dipalmitoid phosphatidylcholine. This is the main lipid, and other proteins are also there. So if you want to make note of it, so dipalmitoid phosphatidylcholine, which is also known as lecithin, which is going to be there. Sphingolipids are there, right? Then we do have surfactant protein from A to E. That is surfactant protein A, surfactant protein B, C, D, E, like that. So these are also there to stabilize it. And please remember, these are released from the Clara cell. Type 2 pneumocytes as well as Clara cells. And the surfactant is produced by the type 2 pneumocytes. What do you mean by pneumo? Pneumo means air, site means cells, so pneumocyte means the air cells. Basically, we are talking about the respiratory cells that are present there, right? So let's move on. So synthesis of surfactant is just going to be there. Then comes the local fibrinolytic system. I told you that they are going to trap the microemboli. Now understand, if they are blocking the microemboli and too many microemboli come, what is going to happen? The pulmonary system, vessel system or blood system itself will be blocked, which is not a good thing. So that's why we should have the local fibrinolytic system, which are going to break down the clot. So please remember that they do have local fibrinolytic system, and that is going to break down. That is going to break down the blood clots mainly. And PAMPs will be taken care of this fat molecules, and air will be, anyhow, it can be excreted out, right? Blood clots which are present or will come as in the form of microemboli. So that is very, very important. The third thing it is going to do is that they will cause the removal of some things also from the body. What things is going to get removal or removed from the body? They help in the removal of adenosine, bradykinin, the acetylcholine, prostaglandin, serotonin, prostaglandin, serotonin, as well as norepinephrine. Right, so I'll just write it completely over here. Prostaglandin. They help in the removal of that also. They will break down. There are enzymes which are present, which are going to break down these things and help in the removal also. Okay, not the complete removal, but they will help it. They will further enhance it or facilitate it. Okay, let's move on further. Right, you're studying the respiratory system. What an inspiration, right? The inspiration that we are talking about is completely different, right? And the inspiration that we are going to study is going to be very, very different. So yes, the main primary function of the respiratory system is none other than the inspiration and expiration. Inspiration and expiration that we are going to study in detail, because understand the volume and pressure on both the things the questions are going to be asked. And I have taken some difficult question today, so you have to answer all these questions. Okay, now Boyle's law, the whole respiratory mechanism, mechanics of respiration, describe mechanics of respiration in brief, or explain the mechanics of respiration. These are the questions which can come in your university exams also. So this is based on the Boyle's law. What is the Boyle's law? Okay, thank you. So what is the Boyle's law? For now, just concentrate on studies. If you study because of that, then no comments on that. Okay, so Boyle's law. What do you mean by Boyle's law? The first thing is, he says that if we keep the other thing constant like temperature and all, so that will give us the pressure is inversely proportional to volume. And that law we are going to apply over here, and let's see how this is going to work for us. Okay, so P is inversely proportional to the V whenever the temperature is constant. There are various laws also. If I get a chance, I will make the video on those, because not for the UG level, but when we do have the DNB exams especially, or the PG exam, they ask about various gas laws which are present or which can be applicable in the respiratory system. So there are many laws, Charles law and other laws, but that is not going to come. I never saw this question in the UG exam, so I am not going to discuss that over here because it is not required for you guys. Let's move on to the respiratory pressures. We have talked about the Boyle's law, so pressures and volumes that we have to talk, right? So first of all, understand that I told you that whenever the pressure is going to be low, so how to remember? There are two things that we are going to talk about. First is the inspiration, second is the expiration. Inspiration means to inhale and take the air inside. Expiration is the opposite process, right? So what you have to note, the point over here is this: S. S means S is going to come in the inspiration, and that's why you can remember the very easy way that inspiration is going to be acting as a suction pressure. It is not the suction pressure. Understand, just for the mnemonic sake I am telling you, right? Suction pressure is usually negative pressure which is going to get generated in our respiratory system, right? So yes, the suction pressure will be generated, suction pressure will be generated. Because of the suction, the air is not going to come in. The air is going to come in via the diffusion. There is no suction pressure. It is not per se the active process, right? Or we cannot discuss whether it is active or passive on the basis of the pressures. Okay, so first thing, it is clear that the air is going to come with the help of the diffusion and not because of any suction, right? It's the suction pressure which is created. If the suction pressure is created, what is going to happen to the volume of the whole thoracic cavity? It is going to increase because the pressure is, that means there is a less pressure, we can talk like that. There is less P. What should happen to the volume? The volume is inversely proportional, and that's why, according to the Boyle's law, the volume is going to increase, isn't it? And that's why if the volume is increased, there is a lot of space and the pressure is low, outside the pressure is high, and because of that, the diffusion of the air occurs from atmosphere inside our lung. Okay, so this is an easy way to understand, right? And what about expiration? Now understand, X is coming, right? If you just tilt that X, what is going to happen? It will look like positive, isn't it? So please remember, during the expiration, we do have positive pressure in our lungs. Yes, many other faculties, they are taking up, they are watching the videos and they are using the mnemonics, and I have seen a few faculties using the mnemonics and whatever I'm teaching over here, but even some of the students will get benefited. I'll be more than happy, right? So I think the knowledge should be distributed, but again, then you have to make your own mnemonics too. So positive pressure is going to do what thing? The pressure is going to increase in this case and the volume should decrease. So when I'm going to compress the chest cavity, what is going to happen? The alveoli will be compressed too, and as soon as the alveoli get smaller, the air will start coming out. So because of that, the pressure, as soon as it increases, the compression occurs in the thoracic cavity, the alveolar pressure start to increase and the atmospheric pressure is still same, right? So comparatively, now the alveolar pressure is higher and we know that the diffusion is going to occur from high pressure to low pressure. So in the expiration, the air is going to move out. Okay, everything we are going to go in the detail. It's just the very simplified way to tell you. Okay. So next is there are different pressures. Let's see, first of all, let's draw a very simplified diagram. Right? Suppose this is the lung. Now what happens? The lungs are covered with two types of coverings. What are the two types of covering? First, which is sticking towards the lung. And what is this covering is called as? Lung is a viscera, that means this is the organ, understand? Okay, so this is the organ and whichever is towards the organ, we call it as viscera, and there is the Latin name for it, and I hope you will answer this. And outside there is one more thing, right? So which is attached to what thing? So suppose these are the ribs. Let's draw in the black. Okay, so suppose these are the ribs, so this is the chest wall, right? This is lung inside, and the red one that I have drawn over here, this is nothing but the visceral pleura. Pleura means the space, right? Which is, yes, very good, Roy, absolutely correct, bache. Wonderful. I want the message that you have got the gold medal, right? I have got some messages, but they say that ma'am, do not post it, so I do not post it. Whomsoever are comfortable, definitely I'm going to post the same comments or whatever message that you are sending me, right? Next is parietal pleura, which is attached to the chest cavity. Okay, chest cavity or thoracic cavity, you can say that. Now understand, these two are not stick together. There is a space in between, and since it is the space between two pleura, we are going to call it as pleural space. Pleural space. Can you imagine what is there inside the pleural space? There is a fluid inside the pleural space. It is going to have pleural fluid. Normally also there is going to be pleural fluid. My question for you is, who is going to secrete this fluid, or from where this is going to come into the pleura? How much is it? It is around 10 to 20 ml. It is around 10 to 20 ml is going to be there. Just give me a minute and tell me from where this is going to come. Chalo, let's move on. Next is pleural space. I'll tell you the answer over here is that pleural fluid is going to get released from, that was my question, that is going to potential space. Yes, that is going to get released from the parietal pleura. Parietal pleura. Okay, so parietal pleura is going to secrete the fluid every day. Now understand, but still this fluid is 10 to 20 ml. That means there must be some system which is maintaining this pleural fluid. It is not allowing it to increase. Okay, now you have to tell me who is going to maintain that. Okay, before going to go towards that point, I'll like to mention about these two pleural properties. Visceral pleura, it is attached to the lung, and lung has a tendency to collapse inward. The lung has tendency to collapse inward. It is like a rubber band, right? And as I told you, there is going to be surface tension which is going to be there in the lungs because of the alveoli and the water layer which is surrounding the alveoli. So it tends to collapse inward. Understand, the visceral pleura is attached to the lung, so it is going to tend to recoil inward. But the exact opposite is going to happen in the chest wall and the parietal pleura. What they are going to do, the chest wall try to recoil outward. Okay, it tries to go outward. But whenever it tries to go outward, what is going to happen? The parietal pleura is attached to it. The parietal pleura is going to go in the different direction. The visceral pleura is going to go in the different direction. What is going to happen? Because their directions are different, definitely there is a vacuum which is going to create in between. Now understand, I have drawn two things. First thing you can see over here is that this is the open space, right? That means suppose all the alveoli are there, and then they are connected with the airways. Airways are connected with your trachea, that is the entrance, and then they are connected to the nostrils which are open, right? That means if it is directly connected to the atmosphere, that means it is the open system. But that is not in the case of pleura. Pleura, in that what is going to happen? See over here, that at the hilum, this is the hilum, right? That is the entry point from where all the vessels will go inside, the airway will go inside, right? All these things. So at the hilum, both the pleuras are going to meet together. And what is going to happen? Very good, Madhivan. So I hope you are Madhivanan, right? I hope I'm pronouncing it correctly. So apology if I'm not. So they are going to connect to the hilum, at the hilum. So what is going to happen? Suppose, for example, I'll give you the very simplified example. So this is a water glass. Suppose I pour the tea inside it, which is a hot tea. What is going to happen after some time? Because it is an open system, the outside temperature is lower because the AC is on, right? So outside temperature is lower, inside of it is going to have higher temperature. But since it is the open system, after some time if I see, there is going to be exchange of energy. That means it is going to have, after some time, the content in this is going to have the same temperature as that of the room temperature. But this is not in the case of this mug, right? So this is a thermos in which if I pour tea or hot water, what is going to happen? This is a closed system. Okay, so this is a closed system, and the content that it is having, it is going to have the same temperature that it is having before also, at least for some time. Why is it so? Because it is the closed system. The same thing is going to happen in the case of lung. So pleura, they are making the closed system and the lungs are making the open system, right? Why I am stressing too much upon this? Because understand, if you have open system, it cannot have. So lungs are like an open system. So they will be having, at the rest, they will be having atmospheric pressure. As simple as that. Because this will be managed to the, or this will be this will come at the equilibrium with the atmospheric pressure. But this is not in the case of the pleural space. So in the pleura, they are connected at hilum. They both are connected at hilum of the lung. So what is going to happen? They act as the closed system. And what is the benefit of closed system? That they can have any pressure. Can you tell me how much is the pressure in the atmosphere? The pressure in the atmosphere is 760 mmHg, but we call it as 0 because that we take it as 0 or the baseline. From there, we are going to talk about positive and negative. So in two terminologies we can talk. What are the two terminologies? Atmospheric pressure, first of all, absolute pressure and relative pressure. The absolute pressure that we have in the atmosphere, absolute means what is the actual value? That is going to be 760 mmHg. Pressure is in mmHg. Okay, and if I talk about the relative pressure, because you are going to see 0, -1, +1, -2.5, these kind of values. Okay, if my lung is having 0 pressure and outside 760, definitely I myself will be collapsed. Okay, but that is not the case. Why? Because the pressure that they are talking about is the relative pressure. Okay, 0 mmHg. Don't get confused because in the books it is going to be different. Okay. Now understand, atmospheric pressure is 0. So at rest, my lungs are going to have pressure which is going to be 0. Pleura, on the other hand, what is going to happen? Because of this property that the visceral pleura want to go out, the visceral pleura, the parietal pleura. Okay, so what we can write it, there is one more property, that since that property, that is visceral pleura, visceral and parietal pleura, they have the tendency to recoil in opposite direction. They go in the opposite direction. What is the opposite direction over here? First thing, visceral pleura is attached to the lung. Because of lung, it is going to go towards the inward. It is going to show the inward recoil. We can write like that, right? And because of the chest wall, the parietal pleura, which is stick to the chest wall, it will try to recoil outward. It shows outward recoil. So they are going in the opposite direction, plus their closed system. That means no air can come inside them. They do have vacuum which is created. Why? Because it is a closed system and they are going in an opposite direction. So that's why volume is increasing of that area, the space is increasing, and that means the pressure is going to decrease. So that's why at rest, and because of that, at rest they are going to have negative pressure. How much negative pressure is there at rest? It is going to be approximately 2.5 mmHg pressure. 2.5 mmHg pressure. This pressure inside the pleura will be called as intrapleural pressure. Easy? Very, very easy. You're going to have the short note on this also, the pressures and volumes, all these things. So they will be asking you what is the cause of the intrapleural pressure being negative. Because it is a closed system, and as simple as that, because of both the pleuras are going in opposite direction, vacuum is created, and that's why, yes, this is going to be the how to make Aditya. At the end I'll explain you that. Okay, so that is going to cause the negative pressure or suction pressure. Okay, coming to the next point. So this pressure is known as IPP. I can write it over here, intrapleural pressure, or this is also known as intrathoracic pressure. T for thoracic. Intrathoracic pressure. Okay, one more pressure that we are having is the intra-alveolar pressure. What I told you about the lungs, lung is the open system, and that's why at rest what it is going to have, at rest it is going to have 0 mmHg. 0 means what? Is it the absolute pressure or relative? It is the relative pressure and it is not the absolute pressure over here, right? So this is going to be the 0 mmHg or 760 mmHg. Now understand, as soon as you start to inspire, what is going to happen? So during inspiration, where the inspiration start, as soon as your oxygen levels, partial pressure of oxygen falls inside the blood, that is sensed by the chemoreceptors. There are receptors which is sensing the chemicals. What are the chemicals? Decrease in the PO2, increase in the PCO2, increase in the H+ ion in the blood. All these are sensed by your peripheral chemoreceptors. For now, just understand these are the chemicals. So this is going to be received. These signals will be received by the receptors, that is known as chemoreceptors. They will be activated and they will send the signals towards the medulla where all our respiratory centers are present. As soon as they are going to send the signals, the neural networks, they will be activated and neural signals will be formed for the inspiration via the eye neurons. Eye neurons are going to get activated. What are the eye neurons? Eye neurons are nothing but the inspiratory neurons. Okay. As soon as they are activated, they are going to stimulate the intercostal muscles. Which intercostal muscle, bacho? Or I can just write, rather than writing intercostal, I can write inspiratory muscles. Okay, they are going to activate inspiratory muscles. There are main two inspiratory muscles which work at the time of quieting inspiration. Now understand that you're sitting right now, you're still inspiring and expiring. And whenever you're going to do exercise or any strenuous activity, definitely at that time also you're going to do the respiration. But there is a drastic difference, right? at the time you are resting, at that time whatever respiration is going on, it's a quiet respiration. That means you do not have to apply much force. Okay, but if I'm doing pranayama, for example, then I'm going to do like that, right? So that means I'm doing forceful expiration and inspiration, which is nothing but the inspiration and expiration, where I have to apply more force. Okay, so I'm talking over here about the normal resting condition. So what are the muscles which are going to activate over here? There are two muscles. First is the diaphragm, and second is the external intercostal muscle. How to remember? Remember, whenever we talk about internal, external, like that, so in goes with X. Why I said that? Because whenever we are going to come towards the flow volume curves and all, over there also, in never goes with in. Inspiratory never goes with the internal ones. Okay, inspiratory goes with the external ones. That is, in goes with X or X goes with in. Okay, in the whole respiratory system. So inspiratory muscles mein external intercostals will be activated, and the diaphragm. What is the contribution of these muscles? Please remember, please remember, the contribution of diaphragm is much more. That means it is 75%. If the phrenic nerve is damaged, the quality of the life of the person is going to get hampered. Why? Because the 75% phrenic nerve supplies diaphragm. If the phrenic nerve is cut in the accident or any trauma for that matter, what is going to happen? The person won't be able to contract the diaphragm properly, and definitely he is going to have the dyspnea. Okay, that is not in the case of the external intercostal. So first of all, let's understand what is going to happen. I'll try to draw this again. Suppose this is the thorax of some person, right? Bilaterally asymmetrical, but it will work for us, right? Suppose this is the diaphragm. What is going to happen? As soon as, suppose this is the diaphragm, as soon as it gets the signal from the eye neurons, it will start to contract, and as soon as it contracts, so this was the normal position, it is going to go down. Okay, so this condition will be something like this. As soon as it will start to contract, it will go somewhere here during inspiration. So what is going to happen to the vertical diameter? The vertical diameter is going to increase. What is going to happen to the other diameters, anterior, posterior and transverse diameter? Let's see. As soon as your external intercostal muscles, they are going to contract. IC means intercostal muscles. As soon as they are going to contract, this is because of the diaphragm mainly, right? As soon as they are going to contract, they will pull the chest cavity . They will pull the chest cavity or move the chest cavity, you can write, in the direction which is, see, as soon as you inspire, what is going to happen? Your rib cage is going to move slightly outward, forward and upward, right? It is this movement which is going to occur. Okay, so there is a dance like this, right? What it is showing? It is showing the inspiration and expiration, right? So your chest cavity is going to go up, forward and outward. So it is going to move forward, upward and outward. And because of that, all the other diameters, they are going to increase. For example, which diameter? Because it is going outward, my transverse diameter is going to increase. So increase in the transverse diameter. Not only that, if I talk about anterior-posterior diameter, since it is going forward also and outward, so that is why it is going to increase the anterior-posterior diameter also, isn't it? So that means vertical diameter is increased, lateral, that is transverse diameter is increased, anterior-posterior diameter is increased. That means the whole chest cavity volume is going to be increased, and because of that, what is going to happen? So the pressure is decreased, and over here the pressure is going to, during inspiration, because of that the volume of the chest cavity is increased, and that why the pressure is going to go down. So there is decrease in the pressure And in the atmosphere there is high pressure, so I'm just drawing a small lung over here. Less pressure is here. This pressure is constant, but now because the lung has less pressure, relatively there is more pressure in the, let's write like that, how much less? It is going to shift only by 1 mmHg, minus 1 mmHg. Okay, so if I have to write in the absolute value, it is going to be 759 mmHg, which is going to be the absolute pressure inside the lung. What about the atmospheric pressure at this point? It is still 760. You are inspiring, expiring, that doesn't mean the atmospheric pressure is going to change. No, it will remain the same, and because of which the gradient is created towards inward, and you can see it is going to go from 0 to -1 or from 760 to 759. As we already know, it always go from high gradient towards the lower ones, right? High concentration to low concentration, high pressure to low pressure, high volume to low volume. So this is how the air is going to enter into the lung, which we call it as inspiration. So if you have to write inspiration or discuss inspiration, all these things you have to discuss. Why? Because now you are medicos and not the layman. So you can see over here in the alveoli what happens during inspiration. This diagram is also given in the genome. You can see that during the inspiration the pressure is going to go towards the -1. Okay, the opposite thing is going to happen in the expiration. What is going to happen in the expiration? That as soon as you start to expire, the opposite thing will happen. Remember that the normal inspiration is active process. Why? Because these two muscles are activated, and that's why this is going to be, these two muscles are contracting, and for the contraction of muscle we require energy, and because of these two muscles, this is the active process. We have to contract these muscles, right? Now understand, I told you that the lung has the tendency to recoil inward. As soon as there is no force which is pulling them outside, what is going to happen? That if I say the oxygen, as soon as the inspiration is going to happen, we'll continue this process. As soon as the inspiration will occur, what will happen? There is going to be entry of oxygen. The PO2 levels will come back to normal, right? Increase in the PO2 back to the normal. As soon as the other parameters are going to go normally towards the normal, what is going to happen? The chemoreceptor will stop working. There is no signal or activation of the eye neurons. If there is no activation of eye neurons, what is going to happen? This process will stop, right? If no activation of eye neurons will stop working. There is no signals, no neural signals for the external intercostal or diaphragm to contract. So the external intercostal and diaphragm start to relax. As soon as they relax, what is going to happen? Definitely, if they relax, they will come back to normal position. As soon as they come back to normal position, all these diameters will decrease. The vertical diameter will decrease. The anterior-posterior diameter is going to decrease. So all these diameter, chest cavity will come back to the normal position. As soon as it comes back to normal position, but during the inspiration we have extra amount of air which came inside, and now we are compressing the chest cavity. What is going to happen to the inside pressure? Because of the air entry which happened during the inspiration, the pressure during the expiration will start to rise. And if we talk in terms of pressures, how much rise in the pressure can be seen? Again, it is going to be changed by 1 mmHg, that is +1, or we can call it as 761 mmHg. 761 mmHg which is present over here, 0 or 760 mmHg which is going to be present. Again, there is a gradient which is formed, and now the gradient is in the opposite direction. Inside the lungs you do have a lot of pressure which is formed, that is +1 mmHg, and outside it is less, and that's why the expiration will take place. Did I say that any muscle is going to contract? No. I said that whatever muscles those were contracted in the case of inspiration. Now they are just relaxing over here. There is no other contraction at the normal expiration, right? So at quiet or rest, quiet, or let's say at the resting inspiration, quiet or resting expiration, no muscle will contract. No muscle is contracting, and because of which it is the passive process. Only inspiratory muscles over here. This happens because of the only inspiratory muscles, they will relax, and the lung or the alveoli will recoil. Alveoli will show the recoil process, and that's why this whole thing is taking place over here. So please remember, the quiet respiration or quiet expiration is passive process, not the forceful one. If I have to do the forceful expiration, for that I have to activate more muscle. For that I have to put pressure, and to generate that excessive amount of pressure I have to contract the muscle. That is going to be active. This is the only process which is going to be passive, and that is nothing but the resting or quiet. Yes. Good evening, Rahul. Very good evening. Yes, you are right. So this is going to be the case. Can I call you S? 47, you are right. Okay. Right. So coming towards, that was about the normal inspiration and IAP, that is the intra-alveolar pressure. So we'll come back to that diagram again. You can see over here, during expiration there is a change which is happening, that is a positive pressure which is generated over here, and that is none other than the expiratory phase which is shown here. Okay, so inspiration and expiration. If in the exam this question comes even for 2 marks, it is going to take only 5 seconds to draw x and y axis. Okay, show that expiration and inspiration and just write the pressures over there. Just draw a wave like that. It is going to take, trust me, it is going to just take 10 seconds, not more than that, but definitely it will increase your marks as well as your presentation, isn't it? Before going to the ITP, let's discuss about the forceful inspiration. Forceful inspiration, that means I have to take extra amount of air inside. For that I have to contract accessory muscles. I have to take the help of accessory muscles. Okay, what are the accessory muscles that are going to contract in this and help me out for the inspiration? So there are double S which is coming. So please remember, there are 2S also, that is the sternocleidomastoid muscle and the scalene muscle which is going to help us. With that there is one more muscle which is very teeny tiny, but still it helps, ala nasal. Okay, this is the ala nasal muscle. If you start to inspire forcefully and see in yourself in the mirror, you will feel that these nails which are going to that flaps are going to open up widely. Okay, and what is the sternocleidomastoid muscle? This is the muscle which is going to go from the sternum over here. So this is the muscle. If I start inspiring, you can see that there is a prominence of this muscle. That means this is going to get contracted. Okay, and the scalene muscles are also going to help them out. So these are the accessory muscles which are going to help us. Not only that, if I talk about the expiration, the forceful expiration, in that definitely I require the muscles to contract, and that is going to be the active process. Inspiration is always active, be that the quiet inspiration or the forceful one. But the expiration, the resting expiration or the quiet expiration is a passive process, but forceful is going to be active process. Which are the muscles which are going to help us? The abdominal muscles, the recti muscles, right? They are going to help us. Not only that, but also the internal intercostal. They are also going to help us in the forceful expiration. Okay, the pressure is going to be much more DLCO, okay, we'll explain that, Rahul. Tomorrow I'll take up that session because I have to cover this now. Okay, if the time remains, I'm going to discuss that. DLCO is very, very easy. The name itself will cover a lot of things. Okay, don't worry. Now let's discuss about this pressures. It is going to be increasing at the time of inspiration. It is going to be very, very low. It can go as negative as -20 to -30 also in the pleura, and it can go up to let's say -6 to -7 also, right? The pressure is going to decrease a lot, and that's why the air entry is going to increase. This is also going to increase over here much more. Okay, let's talk about the ITP and IPP. What was the ITP or IPP? So suppose this is the lung and the visceral pleura both, right? And suppose this was the parietal pleura. Okay, so in between this, whatever pressure is present, this is known as IPP. The intra-alveolar pressure is also called as intrapulmonary pressure. Don't get confused between intrapulmonary pressure and interpleural pressure. Both are the different things. Inside the pleura, pleural pressure will be there. Pulmonary matlab lungs. So whatever, if they say intrapulmonary, inside the lung, we do have alveoli. So it is the similar as the intra-alveolar pressure that they are talking about. Okay, so let's talk about the resting condition. I told you, since they go in the opposite direction, and that's why the negative pressure is going to be generated at the rest. Even at rest, we do have -2.5 mmHg pressure, because the recoil is going to create the vacuum inside, and since it is the closed system, they cannot have the entry inside the pleura, the air entry inside the pleura. What about during the inspiration? Normal or resting inspiration, at that time the pressure will definitely go in more negative, because in the inspiration we say that the pressure should be suction pressure, which is the negative pressure. So in the resting condition, if we are having -2.5, during the inspiration it should go more in the negative. So this is the case. And during expiration, the normal expiration, it will come back to -2.5 again. It will come back to -2.5 mmHg again. Everywhere it is going to be mmHg whenever I am talking about the pressures over here. Don't get confused with the values of Guyton, because in that centimeters of water, that is the unit which is used. Okay, so don't get confused there. The values are a bit here and there, but I am following the Genonka pressure. Suction pressure means negative pressure, which is negative pressure. Abhinav, okay, you're joining late, I guess. We have discussed that. Okay, coming to the graph again, if I show the volume over here, normal volume of the lung during the normal inspiration, that means how much amount of air came inside the lung during the normal inspiration, that is called as tidal volume, which is shown over here. So see, during the inspiration, what they have shown, inspiration and expiration. So I'm going to divide this part like this. So inspiration versus expiration over there. So you can see we have already discussed this pressure is going in the -1 and +1. That means this is the intra-alveolar pressure or intrapulmonary pressure. Okay, this pressure you can see that around -2.5 it is starting, and this is going towards -6 at the end of inspiration. That means this is none other than the ITP or IPP, that is intrapleural pressure. Okay, so intrapleural pressure or intrathoracic pressure, right? So this is the case. Don't get confused between pulmonary and pleural pressure. And this is the volume that they have given over here. This is nothing but the tidal volume. Usually the tidal volume is 500 ml. The amount of air came in the lung during normal resting inspiration. Or you can write in the opposite way, that the amount of air which is expired by the lung during normal resting expiration. Okay, one and the same thing. Definitely when you inspire inside the lung, this is going to increase, and as we start to expire, only that much amount of air is going to go out. Okay, Now, this reason we have already discussed, so I'm not going to discuss that again. Why the ITP is negative? Because there is first reason, oppository coil. And the other, I don't know whether you have given the answer or not, because I asked the question over here that who is going to drain out the fluid. I told you that the parietal pleura is going to secrete the fluid, mucus, everything in the pleura. But remember, if the pressure becomes positive, it is going to be very, very difficult to keep the alveoli inflated. Okay, and that's why we do not want the positive pressure inside the pleura. So there is one more thing, which is nothing but your lymphatics, which are working day in and day out for sucking that extra amount of fluid from the pleura, and so to keep the pleura intact and have that or maintain that negative pressure over there. Okay, so this is going to be the case. Lymphatic is the drainage system, and that is also a part of safety factor. That means even if the pressure start to become positive, or let's say you do have left ventricular failure, the fluid will start accumulating inside the lungs, okay, in the interstitial fluid, not only the pleura, also in the interstitial fluid of this lung. Now, the lymphatics are not only present in the pleura but also in the interstitium of the lung parenchyma, that is going to drain that extra amount of fluid, right? And this is acting as a safety factor. As soon as the fluid content is increased, that doesn't cause the pulmonary edema quickly, okay? Safety factor for the pulmonary edema. Question comes on the safety factor. We have discussed that in the edema chapter, okay? Let's come to its applied aspect, that what if the positive pressure begins to build up in the pleura, let's say that. What is going to happen? There are two things, simple pneumothorax and tension pneumothorax. So listen carefully. I'm going to draw a diagram over here of the lung, small tiny little lungs, right? And let's say in this direction I'm going to draw another lung. Okay, now let's say this is the pleura. But what happened? Someone stabbed a person. Let's say person A got gunshot wound, okay, and let's say person B got stabbed. But there is one more difference that happened in them, is that there is a flap which is formed here. Now, because of this flap, what is going to happen? Understand. First of all, we'll talk about this. The pore is formed over here. So, now the pleura and valve, let's say valve, okay. So, valve in this is absent. And what is the function of valve? One-way flow. And this valve is present over here. I'll just highlight this valve. Now understand, due to this valve, everything is going to change and the person will land up into severe type of diseases, right? So first thing is that since the gunshot injury is there, so air is going to come inside the pleura, right? Now that is not happening in the general case or in the healthy person, right? Over here, the air will start getting entry into the pleura. That means now the pressure will not be negative over here, right? The pressure will start to go towards the atmospheric pressure. The air entry will happen until what time? Entry till the pressure will become, the pleural pressure will become equal to around atmospheric pressure. Now, if it becomes atmospheric pressure, what is the problem? We did have this surface tension beforehand, which is balanced by this negative pleural pressure. But now, what is happening over here in the patient? That there is no negative pressure to balance the surface tension. Now, what is going to happen? That yes, the lung collapse is possible. So, collapse of the lung collapse. ITP is going to be, what is the change in the ITP that can be seen over here? So the second point I'm writing is the ITP. ITP becomes 0 mmHg and the lung collapse on that side is going to be positive. Now what is going to happen in the tension pneumothorax? Tension means there is a tension developed because of the presence of the valve. Someone stabbed the person with a knife or something like that. The pleura, it formed a stab. Now what is going to happen when the air is going to come from outside into the pleura, the flap will open up. Okay, because the air start to come inside from there. So air will start coming or start pushing this valve and start to get the entry inside the pleura. So the entry of this air is possible from outside into the pleura, but as soon as the air start to go out, this flap will revert back and close the pleura. Now because of this, what is going to happen? The air entry is happening because of the presence of valve. Air entry is happening, but air exit is not possible, or air is not able to go out. Now because of that, more and more air accumulation will start. What will happen to the pressure inside the pleura? Will it be equal to atmosphere? No, it will be more than the atmosphere, even more than the atmosphere. The ITP in this case is going to be positive. Not only 0, but it is going to be much more than the atmospheric pressure. More than the atmospheric pressure. Right. Now what is going to happen to the lung collapse? Lung collapse is going to be pretty severe. Severe lung collapse is seen. Okay, because the air start to accumulate and that is going to push the lung inside. Not only the surfactant is acting to collapse the alveoli, now the outside pleural air will start to pushing the lung towards inward, right? So that is going to cause the severe collapse of the lung of the patient, and the person will land up into the difficulties very soon. Okay, so this is about it, and let's just revise the inspiration and expiration. We have already talked about that, and then we are going to take up the question which I want the answers from you guys. Okay, prevent the lung expansion, exactly correct. Okay, so this is the active process. Tell me what are the muscles which are involved in this. Tell me the muscles, fatafat se. We'll move on forward. I want the answers from you guys. First is the normal inspiration and then is the forceful inspiration. Both the things. What is going to be the muscles that are going to be helping in the normal and chalo, batao fatafat. I'm waiting for your answers. I think there is some lag, right? So I'll move on. First thing, the normal, in the case of normal resting inspiration, two muscles are helpful. First is the diaphragm and second is the external intercostal. In the forceful, we need the help of accessory. And I told you there are two S, that means sternocleidomastoid. Scalene muscles are going to help us with that. Ala nasal. What is, why I'm making you write ala nasal? Because if you have to, very good YouTuber. So if I have to talk about the dyspnea, you can just look at the patient and you can say that if he's sitting, he's not doing any extra effort or the strenuous activity, and if his ala nasal are expanding too much, he's breathing like that, definitely that means he is having difficulty in breathing, and this is the sign which you can see, right? And this via which you can just get to know. Yes, you deleted the comment, right? So this is how you will get to know that whether the patient is suffering from dyspnea or not. Okay, Let's move on to the two main movements that you have to mention whenever the question is going to come on the inspiration and expiration. I told you that the rib cages, they are going to move forward, a bit forward, outward and upward. Okay, so the movement is going to be something like this. Okay, so the movement is going to be something like this of the rib cage. Okay, so that's why it looks, if you see it from the side, okay, so if you see from the lateral side, it looks like this. So as the pump is if you have seen this type of pumps, I don't know what type of generation, from which generation you are and you have seen this or not, but this is a pump from which the water used to come beforehand. So this is the hand pump. Okay, so it does contain a handle, right? The movement of the handle, as it is shown in the diagram, it looks as similar to it, and that's why this during the inspiration, this is also known as pump handle movement. Okay, pump handle movement. And if you see that from the other side, that means from the front side, it looks as a bucket handle. Okay, so it is looking as a bucket handle movement, right? So this is also known as bucket handle movement. Everything else is written over here, that this is because of the lateral diameter and this is because of the anterior-posterior diameter. Remember, in the anterior-posterior diameter, you do have P in this. So you can remember that way, that pump handle start with the P. So they are talking about anterior-posterior diameter, right? And bucket handle, it is because of the lateral one. It contains T, but again in the anterior posterior contains T. So just remember P to P, that is anterior posterior diameter if it is increasing, and if you see that laterally, then it is going to be the pump handle. Okay, otherwise it is going to be the bucket handle. Let's move on. Yes, you are right. 47. So expiration mein, tell me the muscles of expiration during normal and during forceful. Normal means it is a passive process, so no muscle contraction. Rather, it is because of the recoil or relaxation of inspiratory muscle, right, which will lead to the recoil of the alveoli at the same time. Okay, so this is the reason. What about the forceful expiration? In that, I told you in goes with X. We are talking about X right now, not your X or my X. I am talking about expiration, right? So definitely the forceful expiration will happen because of the internal. It will go with in, right? The X will go in. It should go in the dustbin, but in, right? So internal, intercostal, internal intercostal. With that, mainly the rectus muscles, the abdominal muscles are going to help you out. In that, the rectus muscles are the main ones. Okay, so at least two you have to remember, that because of that this is going to be happening, the forceful expiration. The pressure can go up to plus 20 to plus 30, that is the ITP pressure. Okay, coming to the forceful inspiration and expiration, we have already discussed, so I'm not going to write that again. So let's come towards its applied aspect. There are two maneuvers which we ask the patient to do, especially when we are checking the autonomic nervous system examination, when we are doing that. So there are two main maneuvers, that is Valsalva maneuver and Muller's maneuver. What we do is, we ask the subject to inspire, but we ask them to close the glottis. That means we are going to pinch the nose and we are not taking in any air inside the lungs per se, but there is an inspiratory action which is happening, right? That is known as the Muller's maneuver. Opposite Valsalva maneuver, we ask whenever someone is going from the flight and they do have earache. Why it is there? Because as you go on the high altitude, the barometric pressure will fall, and because of that there is a tympanic membrane retraction. The eardrum, they go inside, right? So that's why you have to build up the positive pressure inside your this Eustachian tube, which is connected to the lungs again. So in that, what we ask to expire against closed glottis. That means we are not allowing the air to go out, but still we are expiring. It is like this. So if you do that, you will get to know that your mouth and nose, everything is closed. And if you can try the glottis also, that is this expiratory airways, we are going to close that, and you will feel that your tympanic membrane are just popped out. Okay, so that is the Valsalva maneuver, and mainly we do that in the cases where the earache is there to the subject. Some people, they do have higher sensitivity for the barometric pressure, lower barometric pressure for that matter. And yes, yes, 47, you are absolutely correct, right? There is some lag in the video, so I'm getting the comments a bit late. Okay, anyways, chalo. So expiration against the closed glottis, and the pressure can go as high as 100 mmHg, and that's why it tries to come out through the tympanic membrane. And the tympanic membrane, since it is the curtain like structure which is fixed, which is not going to let the air out, it is going to pop up, and that's how the patient will feel relaxed. Even this maneuver, if you do in the cold situation, such as cold situation, that means the fever wala cold I'm talking about, cough, coryza cold, that cold, if you do that, because the escutcheon tube is filled up, your sinuses and escutcheon tube get filled up with the mucus, and that's why you do have pain, lot of pain, right? So that's why if you try the Valsalva, it can give you a temporary relief. Okay, so that is the case. What about the Muller's maneuver? In that exactly opposite thing we are going to do, inspire against closed glottis. Now that means I'm not letting any air to come in like that, okay, but I'm trying to inspire. So in that case, what is going to happen? My body will try to inspire in that matter also, and that is why what is going to happen, that more negativity will be generated, because I'm not allowing the air to come in. My body will try to create more negative pressure. And remember, in the ITP, the pressure can go very negative as minus 80 mmHg, which is going to be much more negative pressure. Okay, so these are the two maneuvers we can use for the testings also, and we can use for the temporary reliefs also. More in detail you are going to go in the ENT, and you are going to learn that there. Okay, let's move on to the transmural pressure. Now understand, I'll just make you understand with the help of diagrams. This is the last thing that we are going to discuss, right? And then we are going to solve the question, but I want you to understand the concept behind it. Whenever I'm saying intra pressures, that means I'm going to measure the pressure inside. That means either intrapulmonary, that means inside the lungs. Intrapleural means inside the pleura. And if I'm measuring the pressure across any membrane, that will be trans. Why I'm discussing those pressures? Because understand, whatever graphs are going to be there, in that case you are going to get confused. Why? Because you'll not see the x-axis, you'll just see pressure, but most of the graph, it is not mentioned. If it is not mentioned intra, that means they are talking about the transmural pressure. Okay, transmural means across the muscle wall, okay, across those walls. Okay, so suppose this is the lung, this purple color is lung as well as the visceral pleura, and red one is the parietal pleura as well as the chest cavity. Okay, so if I measure the difference between these two, okay, let's call it as x, okay, across the lungs. So what should I call it as? I should call it as trans. And since I'm measuring across the lungs, right, so I have to measure, I have to see first of all two adjacent layer, and for calculating I have to do in minus out. What is in minus out? I'll tell you that. First of all, just concentrate over here. I'm checking the pressure across the lungs, so I'm going to call it as trans, because across, through the lung I'm talking, right? So transpulmonary pressure. If I have to calculate the transpulmonary pressure, what formula I should use? I should use in minus out. Whenever you are measuring the trans, I'm going to write the formula over here itself. Whenever I'm going to measure the pressure trans, first thing I have to do is I should get to know the adjacent layer first. What are the adjacent layers? And whenever I need to calculate, I have to do in minus out. So inner layer minus outer layer wala pressure. Okay, so over here if I apply this formula, So what is the outer pressure over here? Outer pressure is intrapleural, or let's say intrathoracic pressure. Inside over here, intraalveolar pressure. Okay, so I'm going to do is in minus out, that is intraalveolar pressure minus intrathoracic pressure. Can you tell me at the time of rest how much pressure is going to be there? Calculate the pressure. What is the pressure at the time of rest and in the alveoli, in the thorax? Okay, calculate and tell me the answer. We are going to calculate that. Till that point, I'll discuss the next thing, that is across the chest wall. That I'm going to call it as the transmural pressure or the transthoracic pressure. Okay, so either we can write there itself transthoracic. I'll just mention over here, I'll just call it as Y. Y is going to be transthoracic. And if I apply same principle in minus out, that means outer side I'm having atmosphere, inside I have ITP minus atmospheric pressure, isn't it? And if I talk about transrespiratory, I'm going to call it as Z. Okay, now Z is the transrespiratory. Respiration, that means whole respiratory system consists of lung as well as thoracic cavity, right? Otherwise the lung would have collapsed without the lung volume of capacities. Yes, tomorrow I'm going to take that, don't worry. Okay, we have covered the pressure. We are going to talk about the volumes also. Okay, now if I have talked about transrespiratory pressure, tell me the answer of this. 47, Roy, where are you guys? You didn't give me the answer of this. We have discussed all the values also, right? So Z is the transrespiratory pressure. For that I have to calculate in minus out. In over here I'm going to take it as IAP minus the atmospheric pressure. Okay, so simple funda over here. The I'll discuss this in minus out, that is IAP at rest. I know that the IAP at rest is going to be 0 minus, I have to minus the, or I have to subtract the ITP. ITP is already negative, isn't it? So what will be the resultant value? It is going to be plus 2.5. And that's why whatever pressure volume graphs you're going to see, you see that they start from the 0. Why? Because they're not taking the intrapulmonary or intrathoracic pressure, and that's why they start with the 0, right? Because whatever pressure you take it as, even at rest, it is going to be plus 2.5. So that's why they take transpulmonary pressure and not the transthoracic pressure. Don't get confused between that. Okay. Whenever we are going to discuss the graph, it is going to be easier for you. We will understand this. Okay. Transthoracic pressure, in this case, what is the formula? ITP. ITP over here is going to be minus 2.5 minus atmospheric. That will be equal to minus 2.5 mmHg. So it is going to be negative, no doubt about it. What about transrespiratory pressure? In this case, what is the transrespiratory pressure? Transrespiratory pressure is IAP, that is going to be 0, atmospheric is 0, and that's why transrespiratory is going to be 0, 2, isn't it? So it is going to be 0, 0, 0. Yes, you are right. 47. Mathivanan. Right. So transpulmonary pressure across the lung. I'll write in the green color itself. Right. Transthoracic means across the chest wall. Across the chest wall. And transrespiratory means across the lung combined with the chest wall. I hope you understood this. Okay, we have time. We can discuss the volume. Somebody requested in the future, we can discuss it right now. Just give me 5 to 10 minutes of concentration. Okay, who just asked me that 47 you are giving me reply, so it is must for you, right? So see over here, volume we'll discuss right now, we do have time. So suppose I ask someone to inspire and expire. What is going to happen? I'll ask them to inspire and expire in the machine which is attached to the pulley, which is known as spirometer. You must have seen the spirometer, right? So whenever the person is going to inspire, the bell is going to come, bell is going to go down because I'm inspiring. Suppose I'll just draw and make you understand. So suppose this is the cylinder which is connected to a tube, which is connected through a tube through which the person will be very pathetic diagram, I know, but he will be inspiring. Okay, so the person will be inspiring through this, and over here this is connected to the bell. Okay, so there is inner cylinder and outer cylinder which is going to be present, and in between what we attach is the bell. The bell will be inverted like this, which is going to contain some amount of air. Okay, and in between, to make a airtight seal, what we are going to do is 3/4 we are going to fill the water, and this here, this bell will be attached towards the pulley. There will be pulley which is attached. It is attached towards the pulley, and this pulley will be giving the graph to me on the kymograph. Okay, so whenever the person is going to take the air inside, this bell will come down, and because of that this lever is going to move upward, and because of that whenever I talk about the inspiration, I don't know why this is shown very okay. So this is going to be upward upward movement or upstroke, which is seen over here. So inspiration is going to show the upstroke. Now whenever he is going to expire, opposite thing is going to happen, and it is going to give me the expiratory thing, that is nothing but the downstroke, which will be seen. I ask the subject to inspire normally, like whatever he is inspiring during the resting condition. I'll get the graph like this. Okay, so inspire, expire, and I'll ask him to inspire, expire again. Now what I'm going to ask him is, I'm going to ask him to inspire forcefully and then expire forcefully. So I'm going to get a little bigger graph because more pressure changes have been taken place over there, and that's why there are more changes which are going to happen, right? And no matter how strongly he is going to expire, that means now no matter how forcefully, strongly he is going to expire, there is some amount of air which is going to remain inside the lung, which is known as RV. Okay, now understand, inspire, expire, the amount of air which can come in, either come in or go out during normal respiration, this is known as tidal volume. The amount of tidal volume is usually 500 ml. 500 ml. What about the extra amount of air that has come in over here? So see, if I draw a line over here, this is the tidal volume. Okay, now whatever extra amount of air, extra, that means I'm not going to include the tidal volume. This is the extra amount of air came into the lung during forceful inspiration, that is known as inspiratory reserve volume. Inspiratory reserve volume. Okay, and the extra amount of air that has been expired during the forceful expiration, that is known as ERV, right? So IRV can be 2.5 to 3 liter, 3.2 also, and ERV is around 1200, 1.2 to 1.3 liters, right? RV is around 1.1 to 1.2 liters. So I'm writing 1.1 over here. Okay, now understand, volume means the single value that we are talking over here. But if I talk about the adding of these two values, that means if I sum up the two volume, it becomes capacity. Capacity is nothing but two or more volumes which are summated together. So if I add up, I asked the subject to inspire forcefully and I counted how much amount of air he can breathe in completely. That means if I measure the TV plus IRV, this will be known as inspiratory capacity. Inspiratory capacity. What about the expiration? If I calculated this plus this, that is the TV plus ERV, that is called as expiratory capacity. Expiratory capacity. Now I wanted to know that after the normal expiration, how much amount of air is remained in the lung. That means ERV plus RV, that is called as the FRC, functional residual capacity. Functional residual capacity. Functional residual capacity. Now, if I have to talk about the volume that is totally inspired and expired forcefully, that means the IRV plus GV plus ERV, that will be called as the vital capacity. Vital capacity. And if I am going to talk about the whole different thing, this is known as the total lung capacity. Now understand, when we start to expire, there is something known as the CV, that is closing volume. When we start to expire, what happens is our lungs have a specific arrangement. And what is that arrangement? So, suppose if I give the diagram like this, the alveoli which are present on the top or towards the apex of the lung, that is going to be bigger. In the middle we do have medium size, and at the end we are going to have smaller size of alveoli which are going to be present there. Okay, so because of this arrangement, what happens is that the alveoli, during the expiration there is a positive pressure from the lungs, and because of that what is going to happen, the alveoli that tends to collapse first or that is tend to close first is the dependent part alveoli. If I am talking about the upright posture, it is going to be the basal part alveoli. Okay, so because of that what happens is the basal part of alveoli start to close first. Now suppose at this level they start to close, whatever part of the ERV remained after that, that means the amount of air remained inside the lung or came out of lung after the closure of the alveoli in the dependent part, that is known as the closing volume, CV, that means closing volume. And if I add the CV plus RV, that will become CC, that is closing capacity. One question for sure comes on this. Okay, so closing volume and closing capacity, very very important. Okay, so whole and soul thing is this one only. Okay, so I'll just remove this diagram. I hope you understood this diagram. Okay, tomorrow I'll just bring the new diagram. This is a very pathetic diagram. I do not want to get it viral. Right, so time for the question. I hope you guys are ready. Most of the people, they just leave whenever the time of question comes, right? Most of the students get scared after solving the questions. Don't get scared of difficult question, rather you should be enjoying those. I always tell you that as many questions as you are going to solve, you are going to get that much pro in the solving questions, right? So I want you to solve difficult question. I want you to solve as many as questions as you want. Okay, so do as many mistakes as you can before the exam and not during the exam, right? So when you tell someone you are nervous about quiz on the respiratory system, right? So what they give the advice as, just breathe, right? Let's move on. Solve this question and give me the answers. Whomsoever are available, just give me the answer. This is the question number 1. Which of the following happens at the end of normal respiration? Normal respiration के end में. Where I have said drooling, Abhinav. Answers guys, फटाफट से. चलो. The elastic recoil of the lung will be greater than that of the chest wall. Elastic recoil will be lower than that of the chest wall. Exactly balances and will be maximum. The elastic recoil of the lung will be maximum. What they are asking? End of the normal respiration. If you remember, because I haven't told you this exactly, because remember, if you know the concept, definitely you are able to apply those concepts and give the answers. If you do not understand the concept, that is going to be a bit difficult thing, right? We have talked about the pressures at the end of normal respiration. That means at the end of inspiration, at the end of expiration, what will be the value? If I draw like this is inspiration, end of inspiration, expiration, end of expiration. So end of respiration, either that be end of inspiration or end of expiration. Pressure is going to be 0, and if I'm saying 0 pressure is there, what is going to happen to the different recoils? Nobody is answering. Why? Is it that difficult? चलो give me any तुक्का तो मारो यार, कुछ तो बोल ही लगाओ. चलो फटाफट. चलो, I'm moving on to the second question. I'm going to come back and check your answers, right? Which of the following alveolar epithelial cells are known as granular pneumocyte? I haven't told you that also. Okay, Krish, I got the answers and I'm sure that you are right. So tell me the answer for the second one, granular pneumocyte. There are different types of pneumocytes which are going to be present, right? So granular pneumocyte. The third question, before going to go towards it, okay, I wanted to show you this diagram for that question. We'll come back to it. Okay. Chalo. The second question is this one. Third question, I want the answer. During the clinical examination of 28-year-old student present with no significant medical history, while discussing the mechanism of respiration, that Indian physician asked what will be the normal intrapleural pressure at the base of the lung during start of the inspiration. Starting of the inspiration, they have asked. They have asked over here. Next question. Chalo, solve this question. These type of questions are going to come in your exam and you have to solve them. No matter whether it is Prof exam or it is INICET, NEET, PG, right? Pulmonary disease, admitted to the hospital due to reduction in the surface tension, right? In the alveoli, leading to dyspnea and productive cough. Which of the type of epithelial cells are primarily damaged? It should be not reduction, it should be increment in the surface tension. Okay, there should not be reduction in the surface tension, it should be increment due to increase in the surface tension or decrease in the surfactant. That should be the question. Chalo, tell me the answer for this. What question it is? Fourth question. Fifth question, the assertion and reasoning type of question. The intrapleural pressure will be negative or positive depending on the extent of quiet respiration. The reason is given that the pressure in the space between the lung and the chest wall will be subatmospheric in nature. Which one of the following is true? Both are correct, assertion and reason. And reason is the correct explanation. Okay, both are correct, but it is not correct explanation for that assertion. Assertion statement is incorrect, but the reason is correct, and vice versa. I hope this is fifth question. Tell me the answer. Only one guy is giving the answer. That too, I got two answers. Chalo, give me the answers. Whatever it is, it may be wrong. Nobody is watching you. Nobody knows that who is talking to me. Okay, give the answers. Try to give the answers. We'll discuss each and everything. Don't worry about it. Next, all of the following statement about the type 1 pneumocyte are true except. Sixth question. Are they flat epithelial cells, large cytoplasmic extensions, majority of alveolar epithelium, lamellar inclusion bodies are present? Again, only one guy is given the answer. Seventh question. Synthesized by the lung. Surfactant, are they synthesized by the lung? Histamine, can it be? Kallikrein, can it be synthesized by the lung? Angiotensin, can it be synthesized by the lung? Tell me the answers. Next question. This is the diagram that I'm going to come back to. In alveoli of the lungs, the surface tension will be directly proportional to the surfactant concentration per unit area, inversely proportional to it. It does not depend at all. So what is the reason of having this surfactant? I don't know. Equal to surfactant concentration multiplied with surface area. Sometimes it's very funny. Okay, Abhinav is also giving the answer. Abhinav, also tell me of which question, because we are discussing many questions over here. Right? Surface tension wala question number is 8. Ninth, intrapleural pressure will be decreased to -6 during inspiration. Inspiration is passive process. Which of the following is correct? All of the following statements, 10th question, are true except. Prevention of pulmonary edema because of the surfactant, does it take place? Phospholipid film formation will be inhibited, lamellar bodies involved, or decrease effect of surface tension. I'm getting the answers and then give it a shot. Okay, so first of all we'll discuss about these cells first thing first. So see over here, there are two types of cells which are present. So suppose if I draw a simple alveoli again, there are two types of cell. First is very flat cells which are going to be present, which are going to have longer extensions. Okay, so these are squamous, simple squamous cells, right? And they are type 1 pneumocytes. They are lesser in number in comparison to type 2 pneumocytes, but understand they are going to cover lot of large surface area, isn't it? And there is another type of cell which is going to be present. I'll just draw it in the blue color or black color over here. So there is another type of cell which will have the projections like this. Okay, what type of cell it is? It is the cuboidal type of cell which is going to be present. Rather than drawing like this, right? So this is the type 2 cell and this is cuboidal. Definitely it is the one which is going to secrete the surfactant, as you guys were telling me. Okay, but look at the sizes of these two. 95% of the surface area of the alveoli is covered by type 1, and they do have larger extensions. Okay, they are lesser in number, but they cover a lot of surface area. What is their major function? Gas exchange. And for the gas exchange, we require lesser, thinner cell through which the oxygen can easily penetrate and go towards the pulmonary vessels, isn't it? If the cell is larger, it is going to take more time, it is going to take more efforts. So that's why we do not want the oxygen to cross a larger cell. And that's why we have simple squamous cell through which the oxygen can easily pass, isn't it? So that is the function. What about the type 2 pneumocyte? Type 2 pneumocytes are going to be covering only around 5% of the surface area. Covers 5% of surface area. And understand what is their major function. They are cuboidal in structure and their function is to produce the surfactant. That is their major function. That is their major function, and surfactant will reduce the surface tension. So very, very important. Surfactant production is by type 2 pneumocyte. Surfactant proteins which stabilizes this surfactant, they are also produced, some produced by the type 2 pneumocyte and some other surfactant proteins, they are produced by the Clara cells. So if there is no option as type 2 pneumocyte, go for the Clara cell. That is going to be your next best answer which is possible. Right, so this is a simple scenario via which you can solve many questions. Okay, so let's see one by one. First of all, over here, see the this is cuboidal epithelial cell. That means they have shown the type 2 pneumocytes over here, and this is containing its own nucleus which is going to have that genetic code to produce the protein which is required. Right, outside for the protein and this phospholipid, that is the surfactant proteins and the phospholipid TPPC mainly, we require the RER, that is the rough endoplasmic reticulum, onto which the ribosomes will be there, right? And then the Golgi apparatus for the processing of it. Now once the processing occurred, there are going to be the lamellar bodies. This is the ciliary bodies and this is the lamellar bodies, into which these proteins will be folded up. Okay, as soon as you require it, what is going to happen, that once you require it, these lamellar body will they will come towards the surface of the this alveoli, and they will form the mesh like structure and they will release this protein on the top of this water layer, and that's how the surfactant protein comes towards it. So what are the lamellar bodies? They are nothing but in which this proteins, that is the surfactants, they are accumulated. Okay, so these are the vesicles, they do have the surfactant proteins. Okay, and these are the PAMs that I was talking about, alveolar macrophages, which help us in the defense mechanism. Okay, all these things are going to be there in the genetic codes are going to be there in the nucleus. Okay, let's solve the question and say tata bye bye to you guys. Okay, you must be waiting for that. So first question, end of the normal in respiration, the pressure is going to be 0. That means both the recoils are going to balance each other. Otherwise what will happen, if the chest recoil is more, your lungs started to expand much more, because we do not want that. So, and the lung recoil, if it is more, alveoli are going to collapse. That also we do not want. So that means at the 0 pressure, these two forces are balancing each other. Okay, so at the end of normal respiration, please remember this is also known as relaxation pressure or relaxation volume and relaxation pressure. The volume which is present at this point is going to be FRC, functional residual capacity, which is going to be there, and the pressure which is present and the functional residual capacity, this is known as relaxation pressure at the end of normal expiration mainly. Okay, at the end of normal expiration. So the answer over here is going to be option C. I got the right answer from Krish. Okay, so yes, exactly balances each other, and that's why neither the lung expand too much or nor it will collapse, right? So this is the scenario. Next, the second question was alveolar epithelial cells are granular pneumocyte. Understand, whenever we do have a lot of lamellar bodies, because we have zoomed it in one pneumocyte and one lamellar body, it looks like that. But there are going to be multiple lamellar bodies which are going to be present in the type 2 pneumocyte, which are containing the surfactant. And when we see under the compound microscope, definitely we will see that these cells contains granules, and those granules are nothing but the lamellar bodies. And that's why I got again the right answer from Krish, that is type 2 pneumocytes which are going to be there, right? So that is the answer over here. Type 1 pneumocyte, they do not contain any lamellas or any other granules. Okay, what was the answer over here? That they have asked you the normal intrapleural pressure. What is the normal intrapleural pressure at the start of inspiration? That means they are asking about the resting condition, right? And we know at the resting condition it is negative. Why? Because they have asked about the intrapleural pressure. In between the pleura we do have, because of the recoil and that lymphatic system, we do have negative pressure even at rest, and that is none other than minus 2.5, right? I have got the answers from Jamal also. Jamal, right? Vijay, please let me know that which question you are giving the answers for. Okay, right. So let's move on to the next question. That is, a 62-year-old man with a history of smoking, COPD patient hai, and the surfactant is reduced, or you can say the surface tension has been increased, right? And damage, that means surfactant production is decreased. Surfactant production is reduced, and if the surfactant production is reduced, definitely it is the option B and B only. If they would have given the Clara cells over here, then we would have thought of all of the above, but again we do not want type 1 pneumocytes over here. Okay, Abhinav, yes, you were right, bache. I got the answer from you when I gave the answer, but maybe because of lag, internet lag, it is there. Okay, so yes, type 2 pneumocyte is going to be the answer over here again, right? Roy, I got the answer, yes. Assertion, it is saying that intrapleural pressure, now understand they are talking about intrapleural. It can be negative or positive depending on the extent of quiet respiration. If I talk about quiet inspiration also and expiration, during inspiration it is going to go in the minus 6, and during the expiration it will come back towards its normal resting position, that is nothing but the minus 2.5. So it cannot be positive, isn't it? So it cannot be positive, and that's why this is going to be the wrong assertion which they have put up. What about the reason? The pressure in the space between the lungs and the chest wall will be subatmospheric. Atmospheric is 0. Subatmospheric means negative. That is true. The reason is true, but the assertion is wrong, right? So both are not correct. Again, both are not correct. The assertion statement is incorrect. That is true because it cannot be positive. It is why? Because they have written it in the quiet respiration. If they would have just write that forceful respiration, then definitely during the forceful expiration it can go towards the positive, but not during the quiet respiration. Okay. So this is incorrect, and statement is correct. So yes, the assertion is wrong, but the reasoning is correct. Okay, and that's why the answer over here becomes option C. Both the assertion and reason statement are incorrect. No, the reason is correct. Okay, yes, Roy, you entered again back, right? So next, all of the following statement about type 1 pneumocytes are true except flat epithelial cells. Yes, they are flat epithelial cells. How come we know that? Because they are simple squamous cells with lot of cytoplasmic extensions. Yes, that is also true. I just have told you that they cover, because of those cytoplasmic extension, they cover almost around 95% of the surface area. They cover the surface area. Majority of alveolar epithelial cells, that is also true, right? Because they are covering the maximum surface area. And what about the lamellar inclusion bodies? Where were they present? Were they present in the this thing, type 1 or type 2? We have talked about type 2. Yes, you guys are correct. Krish 47, Abhinav, you are absolutely rocking. So it is the lamellar bodies which are not present, because they have asked us about the except over here, right? So inclusion bodies, they are present in the type 2 pneumocytes and not in the type 1, right? Let's move on to the next question. Which of the following is or are synthesized in the lungs? We have talked about many things. Yes, Krishna Reddy, you have entered. Very warm welcome, bache. Yes, you are absolutely right. No issues, bache. Okay, so lamellar inclusion in type 2. That is correct. Which of the following synthesized? We have talked about all of them. right? Especially the type 2 pneumocytes. Yes, we know that the surfactants are produced. Yes, I'm getting the answers. Abhinav, think again. Surfactant, yes, in the lungs in total I'm talking about. Surfactant, yes. In the biochemical reaction, endocrine processes, I have told you that the histamine is also formed and it is helping it to remove also. Claricrine, bradykinin, that is also formed. What about the angiotensin? Understand, angiotensin is not synthesized there. Angiotensin is already going to come into the lungs after getting converted from the renin, which is secreted by the kidney, and the angiotensinogen is secreted by the liver. So it is not the role of the lung over here. So that's why my answer is going to be 1, 2, 3. Okay, angiotensin, which is going to get converted via renin, which is secreted by R for renin, R for renal system. That is from the kidneys, it is going to get secreted, right? Angiotensinogen is secreted by the liver, and angiotensin 1, which is going to get converted by the renin, that is going to go in the lung, and via the ACE it is going to get converted into the second one. This is the type 2 pneumocyte, and this diagram is given in the genome. I have taken that from the genome itself, right? You can have that diagram over there also. Okay, let's move on to the next question. In the alveoli of the lung, surface tension will be, tell me the answers. Directly proportional to the surfactant concentration. I told you the surfactant and surface tension, they are enemies, right? They are like a siblings. One doesn't agree and one will totally agree on it, right? There is no reason for fighting. They do not require reason for fighting, right? So surface tension always want the alveoli to collapse inward, but the surfactant is preventing it. Okay, so they are balancing each other. So that's why this is not directly proportional. Yes, it is inversely proportional, right? Inversely proportional to the surfactant concentration per unit area, does not depend upon directly. Okay, equal to. So please remember, surfactant concentration per unit area say inversely. If the surfactant is more the surfactant per unit area, surfactant, lesser will be the surface tension. Lesser will be the surface tension. Okay, right. So I got the right answer from Abhinav and I got the answers from Krish. So next question, second last question. Statement A, intrapleural pressure will be decreased to -6 during inspiration. Is it true? Is it true, bachcho? Inspiration, it's true, absolutely true, right? Inspiration it goes up to -6, that is true. Fine. What about B? Inspiration is a passive process. Is it true, bachcho? Is it true? It is absolutely false. Why it is false? Because inspiration cannot be the passive process. It requires the external intercostal and diaphragm muscles, right? And that's why it is going to be active process all the time. Active process. So that means the statement A is true but B is false, right? So answer over here is option A. I got the answers from Krish, Roy, Krishna. Yes, Krishna Reddy, you're absolutely rocking, right? Next, all of the following statement about the surfactant are true except prevention of pulmonary edema. Is it going to prevent the pulmonary edema? Yes, because if understand the surface tension is more. If the surfactant is not there, surface tension is going to cause the inward force, right? Remember that there are not only alveoli which are present, the pulmonary vessels, they are crossing across it. So because of that inward pressure, the fluid can move inside the alveoli and that can lead to the pulmonary edema over here also and interstitial edema also. Okay, so this is going to cause the suction of the fluid from the pulmonary capillary into the alveoli, and that's why it can be causing the edema. But because thanks to the surfactant, it will reduce that surface tension and reducing the chances of pulmonary edema. So it is one of the safety factor, that is true. Phospholipid film formation will be inhibited. Now, if I'm talking about surfactant, the major component of it is the phospholipid. If you inhibit the formation of the phospholipid, will the surfactant be formed? No, it is not going to be formed. Yes, Roy, you're correct. Right, DPPC that we talk about it. But if you say that the surfactants are going to inhibit, it won't be forming anymore. So this is the wrong option over here. Okay. Lamellar bodies are involved. Yes, they are involved to store the surfactant. That is true. Decrease the effect of surface tension. That is the first thing that it is going to do. So answer over here is going to be option B. Let's move on. I guess we have finished this. Just try to come towards the main theory questions. That is, write a short note on Weibel's airway classification. Mechanism of respiration, you should know. Mechanism of respiration, see for how many marks the question has been asked. If it is asked for 2 to 4 marks, just don't write much. Just complete at least 1 page inspiration, 1 page expiration, and draw the diagrams. If the question is asked for more than 2 marks, at least one diagram, chart, graph, something you have to include in it, right? Then you can start up with the mechanism of respiration. You can start up with the 2 parts, that is inspiration, expiration, and that too you can divide into 2 further 2, that is the quiet and forceful one. Then you can talk about the muscles that are involved and how the pressure and the volumes are going to change according to the concentration, the pressure gradient, not the concentration. The Boyle's law you can mention if it is more than 4 marks, depending on the time you are having, depending on your writing speed, depending on how much information you recall. On all these things you can just write these questions. Then briefly, the non-respiratory function of the lung. Now they can ask you the functions of lung, then you can divide it into, first of all, the charts are very important. The presentation is very important in the profs, especially when the external checks. Understand, he's not just checking your paper. Your paper is one of the 200 or 250 papers. So it should be presentable, more presentable than the others. So your competition is with your colleagues. Okay, so you have to make it more interesting, and he's not having time to 1 hour for one paper. He's just going to go through. He's not going to read all the things that you have written. So 2 things: always make your paper presentable, easier to grab the important things. For example, I made the chart. If the question is, suppose, let's say, explain briefly the functions of lungs. I'm going to divide into respiratory function, non-respiratory. Respiratory mein I'm going to divide it into 2, that is gas exchange and pH maintenance. And then non-respiratory function I'm going to elaborate. Right? First of all, I'm going to just highlight, make a bullet points of it, and then later on I'm going to explain it. Okay, if it is for 2 marks, just make a chart. If it is not, then you have to make a chart first, so that the examiner will know that you know the answer, and then describe as many things you want to write, you write after that. Okay, so it is going to give a better impression. If you do not, you cannot do that much presentation is not possible at that hasty condition. What you can do is take a highlighter and just highlight it, except the green and red color, that is not allowed. So highlight it. Even if you do not have highlighter, just highlight it with the color pencils that you have, right? So find out the solution so as to make the paper more presentable. Explain the different pressures in the respiratory system. We have discussed that elaborately. First of all, you are going to divide it into 2 types: intra and trans respiratory functions. Intra mein you are going to write about intrapulmonary, intrapleural pressures, and in the trans, again 3 things are there. You can write that with all the diagrams. Okay, so that is all. Thanks a lot. Okay. Fine. So you don't have any fear. So you are not finished until you are proud of it. So steady hard, right? And whatever topic you want me to discuss, you can just write it in the comment box. We can take that topic in the next section. Okay, so that is all from my side, I guess. I hope you like the session, right? And yeah, we'll discuss the main things and we'll complete it. Okay, tomorrow is my last session for this month. Let's see when we are going to meet the next. Okay. Distinction, Teju, I would be love to get the message from your side after passing your exam or after getting your results, right? I'll be waiting for your message to me. I'll be waiting for that. Thank you guys. Thank you so much for the attention. Good night. Take care. Bye bye.